Hello my dears champions, I hope that you are doing so good and I introduce you to Midterm Score Booster series where I'll be talking about life process as a first chapter. So what is the basic motto of this particular series? We know that your midterm is coming, that is everyone's midterm might be starting around 20th of this particular month and there are so less days left for you to browse through everywhere to find the correct content in a condensed form. So this is exactly our motto. We are giving you the condensed form of everything so that you don't have to run around everywhere, and this will be for math as well as science. So today it's time for biology and I am your biology educator Jasmine Joy. So let's begin the one shot for life processes. Over here we will be completing the topics, whatever is in there. I'll be giving you a few complementary questions so that you also get to understand things, and also we'll be talking about few experiments, very important experiments that is given in your NCERT. This video could be long, but yes, as I promised, I'm giving you one shot. That means it should be having everything, right? And it should be worth all your time. So let's begin, children. What do you mean by life processes? Basically, life process means all the processes that is happening inside your body which will help you to sustain or maintain your life. Okay, for example, nutrition is necessary for me to maintain my life. Respiration, obviously oxygen is needed and everything, energy has to be found, so respiration is also important. Circulation, because I want to transport around all the important nutrients. Now if one part of my body will not get the important nutrients, will my body be able to survive? No. Then comes excretion. If the toxic waste and everything just gets accumulated in my body, won't it be a trouble? Obviously. So excretion also has to take place. Talking about reproduction is also a life process, okay? But one thing, it is not necessary to maintain life, which is why I colored it in orange color. So most of the things, it is my handwritten notes itself so that you can find it very easily. But yes, and also the condensed form of it. So we might be a bit fast, but we will complete everything that is in there, okay? So I hope you understood that life process means all the processes which are important for you to maintain life. It includes all these processes. Reproduction is not something that will help you maintain life. It will help to for the generations to go on. If reproduction is not happening, probably the generations won't be going on. We humans will become extinct one day, and we do not want that, right? So that is one thing. Now, something is common in all these processes. Can you guess what is that something which is common in all these processes? It's something internal, not something external that you can figure out. But yes, something is common in all of them. Well, that something is metabolic reactions. What is it? Metabolic reactions. We say, in our body metabolism is happening, and for that we need energy. What do you mean by metabolism? What is happening in all of them? Basically, metabolism means there are two parts of it, anabolism and catabolism. Means, in my body, when there are certain chemical reactions being performed, with the help of it, something is being formed, something new is being synthesized, you call it anabolism. And you call these reactions as anabolic reactions. For example, over here, you have carbon dioxide, you have H2O, and with these particular things, I am going to make something new. That is what? Glucose and oxygen. So basically, over here, I am adding something up and giving rise to something new. Right? So this reaction is known as anabolic reaction, and this process is known as anabolism. So in nutrition also something like this will be happening. In respiration also something like this will be happening. Something definitely would be formed. Okay. Now, apart from that, things also definitely get broken down. So that comes under catabolism, and these reactions are known as catabolic reactions. So what is happening? Basically, same thing. This glucose which was there, okay. If now I want to break down this particular glucose, whenever my body needs energy, it breaks down this particular glucose and it gives rise to CO2 and H2O as well as energy. So over here, I'm breaking or cutting things down, which is why I call this reaction as catabolic reaction, okay? Or this process as catabolism. So together both of them, when anabolism and catabolism comes, you call metabolism is happening inside your body, and it is a common thing that is present in all these processes that you see over here. I hope this is completed. Okay, going ahead, this was just the basic information. Now, one very important thing, if you want the PDF for the notes, there will be a link given in the description box. Okay, it will direct you to a page like this, where dates are mentioned, topics are mentioned, that on what dates what topics are going to get covered, and the PDF link will also be there. So you can download the PDF from there. I hope it's clear. Yes. Now let's talk about our first life process, which is nutrition, and this is something that is of your interest as well. What do you mean by nutrition, children? What do you mean by nutrition? So it is a physiological process, because yes, it is happening inside my body. So it is a physiological process of obtaining nutrients. So this is a process with the help of which I obtain nutrients. But why do I need nutrients? Three things: for growth, for maintenance, and for reproduction to happen. If these things are not done, obviously we won't be able to live on this particular planet. So we need growth, we need maintenance, and also the reproduction process should be happening for our generations to continue, right? So basically the process of nutrition means it is a physiological process by which you are obtaining the nutrients. Now nutrition can be divided into two parts. Okay, there are two different modes of nutrition. There are two different ways that organisms take in the nutrients. Okay, one way is autotrophic way and the other one is heterotrophic way. What do you mean by autotrophic? As I have written over here, auto means self, and trophic word comes from trophos, which means what? Nutrition. So here the organisms which comes under this particular category, they are responsible for self-nutrition, that is, they make their food by themselves. Okay, and what do you mean by heterotrophism? Basically same thing. Hetero means others, okay, and trophis means nutrition. Basically they are depending on others for nutrition. Can you think of someone who is autotrophic, and can you think of someone else who is heterotrophic? I'll tell you a clue. The plants, they are autotrophic, they make their own food, and we are heterotrophs. We do not make our own food. It's not like making chana dal and things like that. No, the basic things are being formed with the help of plants only. That is, they are the autotrophs. So we basically feed on them, or we feed on other heterotrophs who feed on them. So it's a loop that goes on. So let's see, if you have a confusion regarding this, we'll get to know that as well. So autotrophic mode of nutrition means these organisms which you call as autotrophs, they synthesize their own organic food from inorganic substances using light or chemical energy. Let me break things down for you. What do you mean by organic? So whenever there is a compound, and in this compound there is a carbon-carbon bond. Okay, for example, glucose, it is C6H12O6, right? So C6 as in there will be 6 C's, right? So here you can clearly see that as a carbon-carbon bond. And also, if not carbon-carbon, CH bond is there, then also this will be known as organic. So glucose has both CH as well as CC bond. So these compounds are known as what? Organic compounds. Now organic compounds are mostly complex. They are not simple things. They are complex, which is why they have CC bond, they have CH bond, and it is very important thing that I am telling you as of now. Okay, now it is being said that autotrophs are those organisms which make this complex things. But with the help of what? With the help of simple things. What are the simple things? Inorganic substances. That means substances that do not have CH bond or CC bond. Okay, so organic was the one which had the CH bond or CC bond, and they have to be complex. But these guys are what? Inorganic. Can you give an example of this, ma'am? Yes, for sure, I can give an example of this. So basically, inorganic means you have carbon dioxide. If you see carbon dioxide, there is carbon and 2 oxygen, right? 2 oxygen atoms. So basically over here, do you see CC bond or CH bond anywhere over here? No. You do not see CC bond or CH bond. That means it is inorganic. Let's talk about H2O. There is excuse me, there is H2O. So basically over here again there is 2 H and 1 O. Do you see CC or CH bond over here? Again, no. So basically these are organisms which use inorganic substances like CO2 or H2 or something else to make organic substances like glucose and things like that. Okay, but they also need one more thing. Either they need light energy or they need what? chemical energy. Either of them is definitely needed over here. So you have to remember, autotrophs are the ones which makes what? Complex organic substances with the help of simple inorganic substances in the presence of what, children? In the presence of light energy or chemical energy. Done? Okay, light energy as in sun's energy, chemical energy as in some chemical reaction is happening, whatever things are being released out of which if energy is being formed, that energy is being utilized to run other things. So that is what you mean by chemical energy. I'll get to that as well. Okay, so as of now, I think this simple thing is clear to you. Talking about heterotrophs, they do not have this property. They do not have this magical superpower of converting inorganic to organic things. They just want organic substances. Okay, they cannot make anything organic. So what do? They just directly consume organic substances. Okay, so do not synthesize their own food but depends on other organisms for food. And what is the food? Some organic substances. And who is making organic substances, children? It is the autotrophs who make the organic substances. So the heterotrophs either feed on the autotrophs or they feed on other heterotrophs, as I said, who feeds on autotrophs. For example, lion eats goat, goat eats the plant. Somehow this particular thing, the middle link, is feeding on the autotrophs itself. I hope it's also clear. So nutrition, it is what, children? It is a physiological process of what? Obtaining nutrients. Why do we need nutrients? For growth, maintenance, as well as reproduction or any other life process to happen. Okay, now nutrition is divided into two parts. There are two different modes of nutrition, two different ways in which organisms find their own nutrients. First thing is autotrophic mode of nutrition and second one is heterotrophic. Auto means self, nutrition means, trophos means nutrition. So basically people or basically organisms who make their own food are known as autotrophs. Organisms who feed on other organisms for food are known as heterotrophs. Now what do autotrophs do? They make organic substances from inorganic substances. Okay, and it needs the help of light and chemical energy. Now one very important thing, it's a question that have come previous year when I was teaching, and that was, there was an assertion and reason given in which they said that the autotrophs make simple organic substances. I'll write it for you, okay? Simple organic substances from complex inorganic substances. Now, people generally do not see the other things simple and complex. They see, oh, organic is there and inorganic is there. Yes, they make food from inorganic things. But you tell me, can organic things be ever simple? No, they are complex. Can inorganic things be complex? No, they are simple. So these small details, could cost you a mark. So be very, very clear about this, okay? Autotrophic mode of nutrition, if you go ahead and talk about that, again you can divide autotrophs into two. One is photoautotrophs and the other one are chemoautotrophs. As you can see over here, plants and everything comes under photoautotrophs. Chemoautotrophs as in there will be certain bacteria, there will be certain other organisms which come in under this particular category. And as the name suggests, photo means light. So these are the organisms, okay, who are using light energy for preparation of their food, that is organic food out of inorganic thing. These are the organisms that use chemical energy for the formation of food. So over here, uses light energy for formation of food. Okay, what do? Simple inorganic things are there. They make complex with the help of light energy. Example, your plants, general green plants, they take carbon dioxide, H2O. In the presence of sunlight, they make it into what? Glucose, which is obviously complex. On the other hand, over here, chemoautotrophs. Now this is something where children get confused, I'll tell you. They use chemical energy for the formation of food, but where do they get chemical energy from? So there has to be some reaction that is happening at first place. So there is some reaction, some chemical reaction that is happening, and with the help of this particular chemical reaction, there will be electrons being released or protons being released, and these protons or electrons will somehow contribute for the formation of chemical energy, which is known as what? ATP. Okay, so basically these guys got the light energy for free. They are not getting light energy for free. They are doing some other chemical reaction with the help of which, they are forming the chemical energy. Okay, it is not a one-step process, it's a very long process where electrons, protons, every one of them are required. So I cannot explain you everything as of now with a limited time, but yes, please understand there is some reaction with the help of which indirectly ATP is being formed. Now, they got ATP and ATP is a chemical energy. This ATP can help the particular organism to now convert simple inorganic things into complex organic things. Ayyo. Into what? Complex organic things. I hope you got it now. Okay, I'll give you an example, it will become more clear to you. Okay, so this is a photoautotrophs, this is chemoautotrophs. Coming to the examples, we have green plants. Okay, you have green plants. There is green algae, okay, which is known as chlorella. It was used to take it was being taken to space and all, like to have nutrition, to provide nutrition for the astronauts over there, because they obviously cannot carry things like that which could easily get desiccated or things like that. So basically, anyways, let's not go in there. I will get deviated, you will get deviated. So there is green algae who do photoautotrophism, and there is photosynthetic bacteria, children. So there are certain bacteria who are not preparing their own food, you call them heterotrophs. There are certain bacteria who prepare their own food and using light energy. Example is cyanobacteria. Cyanobacteria, you also call them blue-green algae. Okay, because they look like they form the algal bloom and stuff like that. So that's why they are known as algae, but no, they are actually bacteria. Now in this particular category comes two bacteria, that is nostoc and anabaena. Okay, there is nostoc and anabaena. If you can try to remember them. Okay, at least have a quick screenshot in your head so that at least if it comes in MCQ stuff like that, you can easily find out things. But mostly, generally, it does not come. Okay, there could be a question like assertion is there, bacteria do not perform photosynthesis. Then that will be wrong. Because bacteria again could perform photosynthesis. It can be photosynthetic bacteria, it can be autotrophic bacteria. So either way, bacteria definitely performs autotrophism, and that will be very wrong to say that it does not perform autotrophism. Okay, so did I say something else as of now before? Anyways, so you have photosynthetic bacteria, two of them, nostoc and anabaena. I hope this part is done. Going ahead, talking about chemoautotrophs, children, there is nitrifying bacteria. If you know the nitrogen cycle, in 8th standard you studied, right? So there were nitrifying bacteria which were converting ammonia to nitrite. Okay, and again, as I said, this is the chemical reaction. This is the chemical reaction which will be giving out ATP indirectly, okay, indirectly, and this ATP will now run the reaction of conversion of simple inorganic thing to complex organic thing. So example is what of nitrifying bacteria? It is Nitrosomonas. Nitrosomonas converts ammonia to nitrite. Whatever energy is there, it is being utilized for the conversion of inorganic to organic stuff. Similarly, there is Nitrobacter, which is converting nitrite to nitrate. So with the help of this, you can solve NSO questions as well. So nitrite, nitrate, bacteria, nitrifying bacteria and all comes. So if you have a little bit idea about all this thing, it will be really useful for you as well. As of now, I won't stress on remembering all of their names, but at least one would work for you to frame a good answer. Okay, if you are asked the difference between phototrophs, chemoautotrophs, etc., etc. So yeah, chemoautotrophs, nitrifying bacteria, there are two of them, Nitrosomonas and Nitrobacter. One converts ammonia to nitrite, other converts nitrite to nitrate. Whatever they do, they give out energy, chemical energy, and this chemical energy is used to drive the conversion of inorganic things to organic stuff. Yeah, and there is something that is known as sulfur bacteria, which will now convert hydrogen sulfide to sulfur. Whatever energy is there, it will help in the other reaction to happen. Okay, so this is about chemoautotrophs. This is the example. You can write it down, not now, as of now, just do all ears. You don't have to use your pen as of now. You will get the PDF. I told you how, right? We will get to know about that once more at the end of the lecture also. As of now, I do not want to disturb you. So will you be able to remember this? Let's do active recall. Active recall as in you are trying to make your brain remember things without writing or making diagrams, nothing. You're not using anything, you're just actively trying to recall. Later on we'll do that. Okay, so this is the example of autotrophic mode of nutrition. Going ahead, children, we have heterotrophic mode of nutrition. Over here, there are 3 different modes. Again, heterotrophs will also have 3 different categories. It's not just autotrophs who can have different categories, right? So, first category of them is parasitic, and you hate them, I'm telling you. So, parasitic mode of nutrition is done by those organisms who derive nutrition from other living organism. So, they are not waiting for other organism to die. They are feeding on a living organism. Okay, and this living organism, for example, me, I am the living organism, then I am the host, because my body is acting as a host for the organism to grow in. Okay, so these organisms show parasitic mode of nutrition, the ones who live on the basis of a living organism, that is the host. Now, there are 2 parasites, there are 2 different type of parasites. One is ectoparasite, as the name suggests, it is there in the external surface. It's not inside our body, it is present externally. And there is something that is known as endoparasite, as in it is inside our body. Okay, it lives inside our body. Examples, ma'am, ectoparasite example that is given in your NCERT textbook is leech. You know leech. Leech comes and sits on the body and sucks in all the blood. Yeah, so you have leech, you have ticks on the dogs, lice on us, and flea also. So all these things are ectoparasite, they do not come and attack us. Now, to not talk about mosquito, mosquito is under a very critical, topic. It is under lots of complication and things like that. So we won't be talking about mosquito as of now. You can do your own research, but yes, you have to remember at least leech, tick and lice which is there in your NCERT textbook. Okay, going ahead, the things that live inside our body, it is tapeworm and roundworm. What is it? Tapeworm and roundworm. Now there is a plant parasite as well, children. In your NCERT textbook, there is a plant parasite. I have shown the picture, you will get to see the picture, but I didn't write it, so I will write it. It is known as cuscuta, or in Hindi you call it Amar Bell. What do you call Amar Bell or cuscuta? How does it work? Everything you will get to know right now. Like after this is being done. Or do you want to see it now? Let me show it to you now itself. Okay. So this is the tapeworm that I was talking about. It is chapta, it is flat, okay. It is just there in the intestine. It goes and creates a ruckus over there. Now, this is leech, okay. This is cuscuta. This thing that you see, the yellow thing that is there on the whole plant, this thing is known as what? Cuscuta. Now, cuscuta, why are they there on the plant? First question. Because cuscuta, as you see, they are yellow in color, they do not have the chlorophyll pigment. Now, if someone does not have chlorophyll pigment, can they do the process of photosynthesis? Even though we didn't start photosynthesis as of now, but let me tell you, for capturing sunlight, okay, you said light energy is required by plants for making food. Now, for capturing the light, what is required? Chlorophyll pigment is required, which is there in the green plants. Okay. Now, and if you talk about green bacteria, they will be having something else, but they definitely have a pigment. So, over here, in the case of cuscuta, this is again a plant, but it does not have enough amount of chlorophyll for absorption of light. So, what it will do, it cannot prepare its own food. If light is not there, can it prepare its own food? No. So, it decides that I will feed on a plant instead. Why to make my own food? I will just directly feed on a plant. So, it just grows in there, takes in all nutrients from the growing part. And how does it take in the nutrients? With the help of this particular structure. So, just imagine this is cuscuta. This cuscuta basically have things like this which gets inside the plant's body. Okay. What are these finger-like projections known as? It is known as haustoria. Okay. It is what? Haustoria. It is storing something. You can remember like that. Okay. It is storing something from the host. So, you can remember haustoria is there. Basically, this gets inside the plant's body, gets into the xylem and phloem, takes in all the nutrients in there and behaves as if nothing happened. They will go flower also over here itself. The cuscuta flowers, they reproduce and the cycle goes on. But yes, you have to remember the name. Name is cuscuta, Amar Bell. And how do they do everything? With the help of haustoria, which are finger-like projections. Okay. Now, it's clear. I hope. Now, coming back to parasitic. So, parasitic mode of nutrition is done, I believe. Okay. Ecto, endo and living host is there, very much important. Going ahead with next one, which is saprophytic. Now, saprophytic, no, so many questions have come from this particular topic. Okay, example of saprophytes and things like that, what kind of digestion they show and things like that. So let's see. Saprophytes are those organisms, or saprotrophs, you also call them saprotrophs. Okay, sapro means dead and decaying. Sapro, the term means dead and decaying. So these are the organisms that derive nutrition from dead and decaying things. So we as human beings kind of hate dead and decaying things, but they kind of love dead and decaying things. They are the exact opposite of us. You can remember like that. Now what is the example, children? The fungi. Okay, the fungi, things that you see, mushroom, all the different bread mold. Okay, so bread mold is also known as rhizopus. Okay, there is different species of rhizopus, but yes, they are also known as bread mold and rhizopus, the same thing. So do not get confused if the term rhizopus comes. Okay, there is a term rhizopus given over here. Please do not get confused if it comes for your exam, because about rhizopus, it is given in how do organisms reproduce. So it's not like something out of NCERT. Okay, and this is the same thing, the bread mold thing only. And mushroom is there. Okay, and also some bacteria. So bacteria is an all-rounder. It comes under autotrophs, it comes under heterotrophs as well. So there are some bacteria which comes under heterotrophs, and they are what? Saprophytic in nature. You can remember FB, that is Facebook, I always tell in class. So Facebook, F for fungi and B for what? Bacteria. Now going ahead, how do things work in there? So basically just imagine there's a dead organism, it's decaying organism, okay? So dead and decaying, so smelly organism is there. There is a mushroom beside it, okay? These mushrooms, first thing that they do is release the enzymes, okay? First thing that they do is they release the enzymes. Now obviously these enzymes will get inside, okay? These enzymes will not get inside, these enzymes will go act on the dead and decaying thing, okay? And these enzymes will break down the things into smaller things, and after that there might be obviously nutrients in there stored in the organism. These nutrients are being uptaken by the mushroom back. Okay, these nutrients are being taken in by the mushroom. So mushroom is also like, see, I won't want to touch the dead organism, I'll just release the enzymes outside. So everything happens outside. So you can remember like this. Okay, it's a very good way to remember. So mushroom is all pretty thing, it does not want to make it dirty. So you might have seen white color mushroom, it's always good looking, right? So basically they don't want dirty things. So they say they release the enzymes, they break things down, they take in the nutrients, and that's all. So this step means they are doing external digestion. What are they doing, children? They are doing external digestion. Digestion is happening outside the body. Digestion means breakdown of food, right? So breakdown of food and everything is happening outside the body. Everything is being taken out from outside the body. So this is external digestion, and this is what makes us different from them. Even though there might be certain organisms, certain human beings who eat decaying things also. But what is that? Something that makes us totally different from them is that we do not do external digestion, not fertilization. So basically just imagine, okay, just imagine I am a human being and there's something food given present over here, and I think that, okay, instead of taking in that food, I'll just release of enzymes. Do you have that superpower to do that? I do not have, and I don't think any human being has that. Okay, so basically that's why we are different, and they show what? External digestion. Please remember this. That is about saprotrophs, fungus and bacteria are the examples. Most important example is a bread mold and mushroom and yeast. Yeast is also fungi. It's the only unicellular fungi. Yeast is also there. Okay, done. It's all your NCERT examples only, nothing that I have added from my end. Going ahead, we studied this. What was the name? Cuscuta, Hosteria. Okay. Organism who break down the food outside the body are, tell me, this is a question for you. Organism who break down the food outside their body. That means outside their body if food is getting broken down, that means what is happening, children? External digestion is happening. Agree? Now, virus, okay, chodo. Virus gets inside the host and then only it works. Tapeworm, again it is inside the intestine. Cuscuta, it is there, again it goes, seeps in the body and takes in and gets inside its body. Things gets inside its body and then it is getting broken down. It's not like that. So the only option that suits our question is A, that is rhizopus. If you got yourself correct, give yourself a good tap on your back. Yeah. Then going ahead with the third mode of nutrition in Holozoic. Okay, there is third variety also in which we come. So Holozoic. Organisms ingest the organic food material and break the food inside the body. So we are those organisms. We humans come under this particular category. We have a burger, we take in that burger, chew that burger, eat it. So I know that you might be feeling to eat a burger as of now, but no, it's not healthy. So basically, you might be seeing nothing is there which is externally happening. Everything is internally happening, which is why we say that they show internal digestion. So if you want to subcategorize Holozoic mode of nutrition, like who all comes under this, you can subcategorize. There are carnivores that feeds on animals, there is omnivore that feeds on animals and plants, there is herbivore which eats on plants, there is sanguivores which feeds on blood and stuff like that. But all of them, they are Holozoic because the digestion is happening inside. It is not happening outside. Am I clear with this thing? Okay, I hope that this is very much clear. Now remember one more thing. Saprophytes, no children, over here, saprophytes, they are also known as decomposers. They are also known as decomposers because they are decomposing stuff. Okay, they are breaking things down, the dead and decaying, whatever leaf and everything is there. And decomposers are very much different. Okay, very much different from detritivores. They also feed on dead and decaying things, but the thing is that they do internal digestion. These guys, they do internal digestion. For example, earthworm, stuff like that. Okay, please do not get confused between decomposers, that is saprotrophs, and detritivores, and that is also a very important question. Okay, saprotrophs are known as decomposers, detritivores is something else. You can remember like that. Okay, done with Holozoic. So under heterotrophs, what are the three? Parasitic, Holozoic and saprotrophic. Parasitic, example, ectoparasite that lives outside, endoparasite that lives inside. Ectoparasite, example, ticks, leech, flea, okay, all this thing, lice. Okay, endoparasite, example, roundworm, tapeworm, all the worms that you can possibly imagine, not all the worms, but yes. Now, if you talk about plant parasite, it is Cuscuta or Amarbel. With the help of what does it get into? Hostoria, finger-like projections. Now, getting into saprotrophs, they do external digestion outside the body. They release out enzymes. Examples are rhizopus, that is bread mold. And going ahead, what do we have, children? We have mushroom, we have yeast, and also some bacteria. Okay, that is why I say FB is this thing, this category. Now, they are also known as decomposers. Decomposers are very different from detritivores. Okay. Now, Holozoic mode of nutrition is, that means that they're taking in the food and the food is getting broken down inside the body. Now, detritivores comes under somewhat this category, not that category. Okay, and they show internal digestion. Done? Okay. Is the AC not working? Okay. Anyways, next thing that comes in is photosynthesis, and now begins the most crucial time. From here, things are going to get more serious. So, you know, photosynthesis comes under autotrophic mode of nutrition. Photoautotrophs does this particular process. Now, as you see, light, photo means light and synthesis means formation. But what are they forming? What is getting formed? The answer is food is getting formed. In what form is food getting formed, children? In the form of glucose, the food is getting formed. But is it stored in the form of that glucose itself? No, it is stored in the form of starch. Please remember this. This is stored, the glucose, basically the food is being stored in the form of starch. Now, whenever the plant is hungry, does not have enough food, they can break down the starch back into glucose and utilize it for whatever process it wants to. Now, here only general information in humans. In humans also the food is stored in the base. In humans it is stored in the form of glycogen. Will you remember this? Glycogen. Okay, glycogen. Fine. So, and the process of photosynthesis, as the plant says, it's very easy. So, we are going to dive into that. Let's see. So, this is the chemical reaction and they will ask you to write a balanced chemical reaction. First thing, there will be two inorganic things that we definitely need, and what are those two things? Carbon dioxide and H2O. Why do I call it inorganic? Because they do not have CH or CC bond. Agree? What are they going to form, children? It is going to form what? Tell me. it is definitely going to form what? Glucose. Okay, and this glucose, glucose is formed, and also oxygen is formed, which is why we say that plants, they give out oxygen, and that's why we should be planting more plants, because they give out oxygen. This is the process through which they give out oxygen, and you'll get to know from where and all oxygen is coming. Now one more thing, some amount of H2O is also being released. Okay, some amount of H2O which was being used, it is being used up, but from somewhere else, from some other process, some amount of H2O is also being released, and in this whole process, obviously, the chemical energy, that is ATP, is also being produced. Okay, so while they are making food, they are producing ATP as well, some amount of ATP as well. Remember that. So what all do you need? You need 2 people over here and 3 people over there. ATP is an extra thing that you should be keeping there. Now, how can you balance things? I won't be talking about the chemistry details, but still. So there is 6 here. This carbon also has 6, so that is sorted. Let's sort it out. Here, before hydrogen, there is 12. So just break this 12 into 6 plus 6. That's all. Okay. So here there is 6 CO2. This guy is totally fine with it. So it's okay. Do not touch that guy. Now over here, H2 is there. It has 12. Okay. There is 12 H2 being there. So we can divide into 6 and 6 over here. As I said, I am not talking about the actual way to balance things, but for you to remember things. Now, this particular photosynthesis reaction will be incomplete if you are not writing sunlight and chlorophyll, because light energy is required, and who is the one that captures this light energy? It is the chlorophyll pigment, children. Okay. Now, does oxidation and reduction happen here? Now you might be thinking, why is ma'am telling about all this thing? It is very important if you want to remember the 3 events of photosynthesis. Okay, I am not here to make you make you mug up things. I am here to help you out with understanding concepts as well. So basically what is happening, children? There are 2 things that happens in the reaction of photosynthesis. The carbon dioxide that you see, it is getting reduced as in it will gain a lot of hydrogen atoms. Okay, hydrogen. So basically CO2, it is getting reduced to what? Glucose. As you can see, it didn't have any hydrogen. Over here it has a lot. Now over here, what happens to H2? H2 on the other hand gets oxidized. Okay, it is oxidation happens to it, and it forms oxygen. So oxygen comes from where, children? Oxygen comes from H2O. Please remember it. Okay, oxygen comes from H2O. Now over here oxidation is happening, that means that it is losing out hydrogen. Now it might not be very important for you to remember this, but as I said, for understanding 3 events of photosynthesis, it will be helpful. So your glucose is made with the help of carbon dioxide. Your oxygen is made with the help of H2O. Okay, so this is undergoing oxidation, this is undergoing reduction. Now apart from all these things that is being required over here, UCH2O, CO2 is being required, chlorophyll, sunlight is being required. Apart from that, there are certain other things which is also being required, which is what? Nitrogen. Okay, the plant definitely needs nitrogen. Why? For making our chlorophyll pigments, it needs nitrogen. There are other functions as well, but yes. So nitrogen is there, children, and nitrogen is taken in the form of inorganic nitrates or nitrites. Again, an important question. Something that you definitely miss, because when you flip around, it's not there exactly in photosynthesis. If you go around, you'll see that. Okay, so basically nitrogen is also utilized, but plants cannot take in atmospheric nitrogen. In our atmosphere, nitrogen is the most common gas, right? But can we take it? We can, but can the plant take it? Basically no. Okay, so instead of taking the atmospheric nitrogen, it is being fixed with the help of bacteria which is there in the roots and things like that, and they take nitrogen in the form of inorganic nitrates or nitrites. Okay, nitrates or nitrites. Do you remember this? Apart from nitrogen, phosphorus, iron and magnesium is also required. So nitrogen, phosphorus, iron, magnesium. NPK you might have studied, okay, but still there is something extra, that is nitrogen, phosphorus, iron and magnesium. 4 things. So all these things are required, definitely these things are also required. Important. Going ahead, so these are all small information which comes in assertion, these are in MCQs. So this is why I am stressing on it. Now children, am I missing on anything? Okay. Okay, fine. So before going ahead to the 3 events of photosynthesis, we have to talk about the story of the leaf and the chloroplast. So when a leaf is there and it is being cut, now if you see the side view of the leaf, okay, if you are seeing the view like this is being cut, and this is a view, that is the inner view of this, right? So over here also it's being cut, I don't know how will you understand this, but yes, I am trying to make you understand. Anyways, when you cut this, you see the inner view. Okay, in the inner view, when you see, it looks like this. Now, I don't want to make this slide dirty, so I am just removing that. Okay. Now, again, this picture is given in your NCERT, and it's very important for you to notice the picture diagram. Okay. So there are certain cells at the upper layer. You call them epidermal cells. What type of epidermal cells are they? Those upper epidermal cells. Array, just a second. Yeah, these are upper epidermal cells. Agree? Now above it they have cuticle. Above it they will be having cuticle. Below also they have lower epidermal cells. Lower epidermis I will write, okay. This layer is known as lower epidermis, and there is cuticle again. Now the topmost layer, which are closed cells, okay, they are close to each other, you call them palisade mesophyll cells. What do you call them? Palisade mesophyll cells. Is this important, ma'am? Very much. Now below it, if you see, they are loosely arranged cells also, which you call as spongy mesophyll cells. Now you see these structures, this is the xylem and the phloem, that is the vascular bundle, okay. Xylem and the phloem together they form the vascular bundle. Vascular bundle as in they are responsible for transporting things in and out. That is none of our concern as of now. Okay, what is our concern? Palisade mesophyll and then spongy mesophyll, and also in the lower epidermis as well as the upper epidermis you will be finding things like this, which are known as guard cells. These guard cells are guarding a pore which is known as stomatal pore. Okay, and why am I very specifically writing all these things over here? Because children, there are so much number of chloroplasts in these cells, and chloroplast is the organelle which stores the chlorophyll pigments, and without chlorophyll pigments photosynthesis cannot happen. So these guys have large amount of chloroplast. Now how does question come? Which among them have the most amount of chloroplast? So the first position is given to palisade mesophyll. Okay, it has the most amount of chloroplast. Then comes spongy mesophyll, and then comes what? Guard cells. Done? Done? So if a question comes and they are marking 1, 2 and 3, which has happened before, you have to say in order which one has the most. It is 1 and 2, not 3. 3 comes last. Okay? But they definitely have. Guard cells also have chloroplast. Now if you see the structure of chloroplast, how does it look? Let's see. So over here they have two membranes. One is the outer membrane. This is the outer membrane. This thing that you see over here, this is the inner membrane. So they have two membrane. Okay, they have intermembrane space as well. So I'm not writing the whole thing. And then they have this coin shaped structure. Now one coin shaped structure that you see over here, it is known as thylakoid. Thylakoid is visible. One coin shaped structure is known as thylakoid. When you take it as a stack of structures, all this as a whole, what do you call it? Granum. Granum. Okay, so this is one granum, this is another, this is another. Now, one more thing, one more very important thing. This space that you see over here, what do you call it? Stroma. The space that you see over here is known as stroma. Why is ma'am telling about all this thing? Because to understand the event of photosynthesis, you might need reference of all these things that I have started with. Okay, so this khali area that you see over here, it is known as stroma. It is the sap that is there. It is filled with lots of enzymes and stuff like that. And over here you have thylakoid, you have stack of them, that is known as grana, and then there's something that connects both of them. You call them stroma lamellae. Stroma lamellae. So do not be confused between stroma lamellae and stroma, that is different. Okay. Now thylakoid is the place where you will be finding the chlorophyll pigments. So granum has so many thylakoids, so they will also have lots of chlorophyll. Stroma lamellae also have chlorophyll pigments, but stroma it does not have. Okay, so remember thylakoid has, if thylakoid has, obviously grana will be having, and the connecting link will also be having, otherwise they cannot be friends with them. You can remember like that. And something that is extra outside, that is stroma. Okay, I hope this is clear. So out of this you have to understand who has the most number of chloroplast now, or the chlorophyll. If you have more number of chloroplast, that means you have more number of chlorophyll as well, right? Now if you talk about the chloroplast, this is the structure, and now we go ahead with the 3 events of photosynthesis, which is very much important for you. Do not mess up with the events, number them properly. You need to understand that. Okay, so the whole photosynthetic reaction I can divide into 2 parts. One is light reaction and the other one is dark reaction. Okay, dark reaction I also call it Calvin cycle, because Calvin was the guy who discovered this. Okay, so that is Calvin cycle. Now why it is known as light reaction? Because this particular reaction, this set of reactions to happen, it needs the presence of what, children? Light, which is why we say it is light dependent. Dark reaction means it happens only in dark. That is a common doubt. No, it does not happen only in dark. It can happen even in dark. That is the thing. So it is light independent. It does not need light. If light is there, it's okay. It can happen with or without light. Please frame your sentence properly. Okay. Now where is the reaction location? Where does it happen? The light reaction and everything. In the thylakoid. What was the thylakoid, children? This coin shaped thing that you saw over here, it was the thylakoid. Now if you talk about dark reaction, where does it take place? In the stroma. That was the extra space that was surrounding the thylakoid and granum and everything. Will you forget this? No, you do not have to forget this. Okay, this is the dark reaction, this is the light reaction. Now the first event of photosynthesis. So plant is there. The first thing that it does is it tries to absorb the sunlight with the help of what, children? With the help of the chlorophyll pigments that are being present over there. Okay, with the help of the chlorophyll pigments, absorption of light energy takes place. Now, can the plant use this particular light energy for its own purposes? No, it has to convert this light energy to chemical energy. So absorption is done, now conversion has to be taken place. So the next step will be conversion of light energy to chemical energy. What is chemical energy? Basically ATP, NADPH. These are chemical molecules which stores energy in their bonds. So ATP means adenosine triphosphate, means three phosphate atoms are there. So whatever energy is being stored in this bond, okay, when this particular molecule is being utilized for some reaction to happen, they break these bonds and the energy will be released, which will be helping to run the whole reaction, which is why we say that it has lots of energy. This molecule has lots of energy. So the light energy is being converted to chemical energy, and what is chemical energy? ATP and NADPH. Okay, one more thing happens simultaneously. When the light falls, when the light is being absorbed falls, the H2O which was there, remember the H2O, and I said H2O is responsible for formation of oxygen. The H2O will get lysed, it will get broken down to hydrogen and oxygen. Okay, and this process is known as photolysis. Why photolysis? Light is there and this light is responsible for lysis, that is breakdown. Photolysis of what, children? H2O. Okay, photolysis of water or H2O to give rise to what? Oxygen. So this is where the oxygen is being released. So you saw how H2O is being utilized in photosynthesis reaction to give oxygen. But we didn't study yet about carbon dioxide giving rise to glucose. When will that happen? That will happen now. So basically, why did we do this particular process, this light reaction? Because we wanted chemical energy. Okay, we wanted chemical energy to be there. That was the whole purpose of it. Now this chemical energy is now utilized, okay, for the conversion of, or basically reduction of, carbon dioxide to carbohydrate. That is, this particular reaction to occur, it needs chemical energy which is made in the light reaction. Agree? And this is the third event, that is reduction of carbon dioxide to carbohydrate. What is the first thing? Absorption of light energy. Second thing, conversion of light to chemical, and along with it one more thing is also happening, photolysis of water, which is very important. Where does oxygen come from? From photolysis of water itself, from the breakdown of water. Going ahead, what do we have? The next step is what? Reduction of carbon dioxide to what, children? Carbohydrate. For this, what is needed? Chemical energy is needed. So can this happen without this reactions? No. It cannot happen with the light, without light reactions happening. I hope this is also clear to you. Yeah, there's a question for you, I think. Next. Yes, if the plant doesn't receive light energy, will dark reaction happen? Can it happen? You only think. If light energy is not there, how will chemical energy be formed? For some time the plant might sustain, but how? For a long run, obviously it needs chemical energy. Obviously, it needs chemical energy, and for chemical energy, needs light energy's conversion to chemical energy. That means light reaction has to take place. So the answer will be a big no. Okay? Going ahead. Does these steps occur one after the other in all plants? So in all plants, does it happen like that, like one this is done and then carbon dioxide energy is given for the reduction of carbon dioxide to happen? It happens in daytime and nighttime and things like that. Is it like that for every plant? The answer is no. Okay, so as you can see over, this is the cute cacti over here. Plants like cacti perform a special form of photosynthesis which you call as CAM pathway. What do you call it? CAM pathway. Now CAM also stands for crassulacean acid metabolism, because this is being formed over here. Anyways, we don't have to dig our head in that. As of now, just remember they are doing CAM pathway, and what happens? What is so unique about giving a new pathway and putting a name for it? Okay, let's see. So basically, you know that cacti are found in very dry regions. So in these dry regions, in the morning time, if they open up their stomata for taking in carbon dioxide, because carbon dioxide obviously is necessary, right? So you need a stomatal pores to open up and to take in carbon dioxide. But when they open stomatal pores, what will happen? H2O is also being released outside. Now that is one big problem. Can we avoid that? Yes, we can avoid that by doing one thing. In daytime, close the stomata. That's the only option. In daytime, close the stomata. So that means in daytime, light reaction will happen. Okay, because nothing has to be taken from outside, only light energy has to be taken. Light energy is being taken, chlorophyll absorbs it, converts into chemical energy and keeps like that, no problem. But in daytime, conversion of carbon dioxide to carbohydrate does not happen, because carbon dioxide cannot be taken in. Okay, so what is the next option that we have? At night time, open the stomata, take in whatever, how much ever carbon dioxide you want, and store it in a different form so that it does not undergo any set of reaction. It is in intact form. Okay, so basically what happens at night time, stomata opens. At night time, stomata it opens. Carbon dioxide is taken in. Carbon dioxide is converted to something that is known as malic acid or malate. This malic acid in the morning time, when there is enough amount of ATP already being formed, is now converted back to CO2. Okay, it converted back to CO2, and now the whole reaction can take place. I hope you got this. So at night, CO2 is fixed and stored. That is the only purpose. So here the things are different, because CO2 absorption is not happening every time. It is only happening at night, and it is getting fixed, stored, and then later on this malic acid is getting broken down to CO2 again, and the whole reaction continues in the morning. So in the morning, with the help of light energy, photosynthesis, it continues. This is all about CAM pathway. Okay, or CAM pathway. So every time it's not necessary that all the reaction occurs very simultaneously. It can occur in a different time as well. Done, children? Okay, so there are other pathways as well, C4, C3, lots of things, but we won't be digging deep into that as well, because for your boards it's not necessary. Okay, let's talk about stomata guy now, since we were talking about how carbon dioxide was taken up, right? So let's see. So this is a guard cell. These are the guard cells. These things that you see are what? Guard cells. And here is the stomatal pore, which is closed as of now. Stomatal pore. And surrounding it, surrounding the guard cells, there are certain cells which are known as subsidiary cells. Subsidiary cells. As in, they are very dear. They are just subsiding them. They are just near them. Okay. Now children, what happens? Basically, when enough amount of light is there, or when low CO2 is there, when low CO2 is there, this has to open up, right, to take in more CO2. So when enough amount of light is there, or when low CO2 is there, basically let's choose light over here. Okay, the light receptors which are there on the cells, it becomes active. Now when light receptors it gets active, what happens? There are certain proton pumps. What do you mean by proton pumps? The pumps that could let out the protons outside. okay, or inside. So protons means H plus ions, okay. So what happens initially, the receptors got active because of enough light, okay, and now the proton pumps are active, and these proton pumps makes the hydrogen move outside. When hydrogen ions moves outside, they are positive ions, right? Now inside it becomes 2 negative. So to compensate that, what happens? As H plus moves outside, K plus ion gets inside, okay. What happens? K plus ions gets inside, and they are like, oh, K plus ion was just waiting for getting inside. It got inside, it becomes a huge ruckus over there. There will be so many ions, positive ions over there, such that basically negative, positive, everything will be there, such that it increases the solute concentration. What do you mean by solute? For example, there is a glass. If I put water, that is the solvent, okay. Inside the solvent, if I put something, that is the solute. So basically over here, this particular K plus ions and every ions get in, and now the solute concentration is way too much. Now what will happen? The water which is there outside near the cells, these water molecules will get inside, because they have to compensate, right? So solvent is more here, solute is more here. So to compensate that, the solvent gets inside. So now the water gets inside. Now you only think, if there is a cell like this, now water, when it gets inside and this area are attached, this area is not attached, it will slowly swell up and become like this. So now this stomatal pore is finally what? Open. I'll repeat once more. Light receptors are active. This makes the proton pumps active, the protons moves outside. Now it's too much negativity inside, so basically the K plus ions, which are positive, they get inside. Lots of K plus ions, a lot of other ions also, they are also there inside the guard cell. It becomes very hypertonic over there. That means the solute concentration is very much over there. So water molecules comes as savior, get inside to balance things out, okay. And when they do, the guard cells, they become swollen. When guard cells, they become swollen, you say that this is in turgid state. You might have studied this in the 9th grade, right? So it becomes what? Turgid. And the stomata, it opens up. Okay? It opens up. You can remember like that. That's about stomatal opening. Please remember these important terms. Guard cells has to be swollen. That's when stomata will be opening up, or else it won't. So these conditions are very important. Okay? Turgid, stomata open, and stuff like that. Going ahead. Now when does it close back? So there is no light, there is no excess, there is excess of CO2, there is no need of taking CO2 inside when things like that happens. Now the receptors are not active, and there could be other hormones, other stress hormones which would be active, but would now lead to what? Say that, hey, K plus ions, go outside. We do not need you now. So all the K plus ions, they move outside. When they move outside, the solute becomes more outside. So the water molecule which just came because of this K plus guy has now to move back again. The water molecules are being lost back again. Now when water molecules move out, obviously the cell will shrink. When the cell is shrunken, the stomata, it will be closed. So remember, guard cell shrinks, that is in the shrunken state, and that's when the stomata closes. Okay. Is it clear to you? Please, very important thing is K plus ions. What are involved over here? K plus ions and water molecules are involved for opening and the closing of stomata. Okay, this is very much important. K plus ions and water molecules. Very important for opening and closing of stomata, and also the states, shrunken state, closed state, whatever comes in. Okay. Okay, now with the help of 2 experiments, we are going to complete the whole thing. Let's see. So let's see the experiment for carbon dioxide. Actually, carbon dioxide is necessary or not, people try to find it out. Let's see how. So there's a plant over here which is kept in a glass container, and in the same glass container over here there is a beaker having sodium hydroxide solution. Let's change the sodium hydroxide to potassium hydroxide. Works? Either way it works, okay. Sodium hydroxide is also doing the same function, but oh my god, I would have fallen here. Potassium hydroxide is most commonly asked. Potassium hydroxide, children, you also call it as KOH. Okay, so it is kept there, and there is something else also. Soda lime is also there to absorb the carbon dioxide. But on the other hand, there is another plant. Okay, here water is there, here potted plant is there, there is light, there is enough air, everything is good over there. Okay, usually you can just cut this out. This is not very much important. Okay, now see, which among them will be able to survive? You tell me. You tell me which among them will be able to survive. I'll tell this will survive more longer. Why, ma'am? Because potassium hydroxide or sodium hydroxide will take in the carbon dioxide. Will absorb the carbon dioxide. Their whole job is to absorb the carbon dioxide. And if carbon dioxide is taken in, can photosynthesis happen, children? Can the plant make their own food? Will it survive? No. On the other hand, if I look over here, everything is good. Everything is basically good. So as of now, light is there. Light is there. The reaction is happening. Yeah. And the CO2 is being, obviously, for example, if light is there, okay, the reaction happens, there is oxygen and everything is there, or everything is set up, okay, there is no problem. When the respiration process also happens, respiration means oxygen is there, oxygen will be breaking down oxygen, and when glucose, when food is being produced, okay, when light is there, food is being produced, agree? Initially it has some amount of carbon dioxide, right? Obviously it will have some amount of carbon dioxide is there. So basically this glucose and glucose is formed. So glucose and oxygen reacts to form carbon dioxide, H2 and energy. So this carbon dioxide again is being utilized for the process of photosynthesis, and this reaction keeps on going. If you are not able to understand, I'll make it out easy for you. Listen, it has light, okay, this light energy, and initially it has carbon dioxide as well, okay. So it makes glucose. Now the glucose plus oxygen which is being released comes together and forms CO2 and H2. This process known as respiration. So plant is doing both photosynthesis and respiration. In this process, in respiration process, CO2 is a byproduct. So this CO2 again can be utilized by the plant to run the whole reaction. But here CO2 itself is not there. So can it run the whole reaction? No. Which is why we say that carbon dioxide is very much important. If you see KOH, please understand that photosynthesis is not happening because CO2 is being taken up. Now there can be another instance when you are asked, there is a leaf being taken up from this container and then it is being checked, okay, whether it is being producing starch or not. Iodine is being put on this particular region which is affected by KOH, okay. So this is the situation where there is a jar of KOH and this part of leaf is there in the KOH jar, okay, and then they are taking this particular part of the leaf and then putting iodine in that to check if starch is being formed. No, starch is not being formed, because to starch to form you have to form glucose first. For glucose to form you need carbon dioxide. You do not have carbon dioxide. Carbon dioxide is taken up by KOH. Then how can you form the starch? You cannot form the starch over there. You cannot form the glucose over there. Glucose can't be forming starch over there, which is why the iodine test fails. Okay, I hope you get it. Going ahead, one more experiment is there. Okay, there is variegated leaves as well, means there will be patches where the chlorophyll pigments will be comparatively lesser or it won't be there. So basically what happens, they take the variegated leaf, they treat it with, first of all they boil it in boiling water. Why do they boil it? To make the cell break down all the membranes around it, so that any solution which will be added up later on will show its effect. Okay, if everything is intact, obviously if we are putting some solution on it, it won't be showing any effect. So that is one thing. Why do we boil? After boiling, okay, there is one more thing being done, I forgot. It is treated with alcohol. Why is it treated with alcohol? You want to damage all the chlorophyll pigments over there, because if chlorophyll pigments comes in between, it won't again show the correct reaction, which is why it is treated with alcohol. Later, now it is done, starch test is done. So first thing that they did was, they had a leaf, okay, they made the leaf, basically they destarched everything, okay, there won't be any starch. You keep the whole plant like that for some day, okay, you keep the whole plant for some day without light you keep it, so that whatever food was there, obviously it will be there over here, but over here there won't be anything, obviously. Why? Because you won't be having chlorophyll pigments over here. This is variegated leaf, right? Now you do the whole reaction. At the end of it, when you put iodine solution, what happens? Wherever starch is there, it will turn into blue-black color. Now wherever starch is not there, it will be remaining colorless. So that is the whole funda of this. That means chlorophyll pigments is also very much important. Okay, so the green area had the chlorophyll pigments, but the other area didn't have the chlorophyll pigments, and since chlorophyll pigment was over here, that is why starch is being formed, and since starch is being formed, that is why it gives a blue-black color. Done with this thing. This is another experiment for chlorophyll. Now time for a question which came the previous year. In the given diagram of a closed stomata, okay, 1, 2, 3 and 4 respectively are. Now obviously you cannot clearly say 1, 2, 3 I guess, but this is how I got it. So I can just give a clear difference. This is something like this, there is something like this, okay, and yeah, this arrow is pointing towards this arrow is pointing towards this. Now you tell me, can you find the answer for this? So what is 1, children? What is this whole thing first? What is this whole thing? That is, what is number 4? Number 4 is guard cell. Let's see, number 4 is guard cell. So this is also could be correct. Now let's see the next one. Number 3. Sorry. Just a second, okay? I marked it wrong. Where is, okay, red color will be visible. Okay, number 3 is this, okay, number 2 is something over here. Now you tell me. So we said that number 4 is anyway guard cell, that is, let's keep it like that, okay. Next thing, number 3, what is it, children? Number 3, it is the vacuole. So number 3, vacuole. So this is again correct. Going ahead, what do we have? Number 1, what is it? It is the nucleus of the whole cell, which is sidelined because of this particular guy, that is a vacuole. What is number 2? It has to be chloroplast because no other options matches the choice. So the correct answer to this question is I remember children complaining about the diagram quality in the question paper, but yes, this happens, so you have to use your common sense there. So this is the correct answer. I hope most of you got it correct. Going ahead, children, what do we have? We have nutrition in amoeba, but I want you to take a break, a quick little break, okay, and just try to recall whatever you have tried to learn, and I will also have a water break as well now. Okay, so I hope that my little chutkus are back from the break, and over here we have nutrition amoeba. From now on we will be talking about human beings as well. So let's start with nutrition amoeba. First thing, you know there's an amoeba, it is unicellular protozoan. What it is, children? Unicellular protozoan. Now they are asking you explain the digestion in a protozoan. And you're like, what is a protozoan? So this is the protozoan that we were talking about. So unicellular protozoan, they are eukaryotic, they have the whole nucleus and everything there intact, but they are unicellular. So this is the nucleus and this is the lysosome, which has all the enzymes, and this is the food. Now the amoeba goes in, takes in the food with the help of something which is known as pseudopodia. I have mentioned over here, these are false feet, okay. So now first process is taking in a food. What do you call this process? As ingestion. So basically there are 5 steps that you need to remember. 5 steps, names of these terms you need to remember. Ingestion, you are intaking something, okay, that is the first step with the help of pseudopodia. Second thing, digestion. I have given you a note so that you can frame your own answers as well. Digestion is where the food vacuole which is formed. So over here when pseudopodia takes things in, it will now form a food vacuole over here. These food vacuole will now fuse with the lysosome, which has all the enzymes. When the lysosome fuses with the food vacuole, it gives off all the enzymes to the food vacuole, and whatever food is there, it gets broken down. This process of digesting the food material is known as digestion. Simple. So after ingestion, lysosome went and infused there, released all its enzyme. Now the food is getting broken down inside the food vacuole itself. This process is known as digestion. So now it will look like this, where the food vacuole have the crumbs of things, okay, undigested food, waste material, everything is there. The next step that happens is absorption. What do you mean by absorption? Whatever important nutrients are there, since this is just a single cellular organism, it does not have organ system to give away everything to all the different parts. It just have to give things to the cytoplasm. So this process where things are being nutrients, important nutrients, not the waste material, okay, are being taken up by the cytoplasm. This is known as the diffusion of nutrients from the food vacuole to cytoplasm is known as absorption. Now children usually get confused between absorption and assimilation. It is different. For example, I am taking something in my hand. This is absorption. Now I am utilizing this to throw it on someone. That is assimilation. Basically, I am utilizing this for some purpose, even though it's not a right example, but still. Okay, this is how you will be able to remember. So assimilation is the process of utilization of nutrients for various metabolic activities. So this unicellular organism also have metabolic activities. So when the absorbed nutrients are being utilized for these metabolic activities, you call this process as what? Assimilation. Okay, and the last process is basically after you're done with it, the waste material which is still stored in the food vacuole, the food vacuole comes and joins in there and gives out all the waste. This process is known as egestion. So what was the first thing? Ingestion. Second thing, digestion. Third thing, absorption. Fourth thing, assimilation. Fifth thing, egestion. Okay, egestion as an exit. E for egestion, you can remember like that. Going ahead, if we talk about paramecium, the same thing happens within their body, but their body structure is different. They have a specific area for taking in food and giving out food. It's not like amoeba. And they also have cilia. This cilia, for example, some food is there. This food is being rolled up when the cilia moves up and down. And the food gets into something like this. This is known as oral groove. Children, you need to practice the diagram as well. Okay. So this is the oral groove. From the oral groove, it reaches the mouth, that is the cytostome. If you do not want to remember it, it's okay, but try to. Okay, cell mouth you can remember. Okay, cytostome, stome as in mouth. You can remember like the cyto as in cell. So this is there. Now this is the food vacuole. The food gets in the food vacuole, and the same set of reaction happen. Lysosome comes in, fuses, ding, ding, and at the end, the food materials goes out with the help of cytoproct. What is it, children? Cytoproct, which is known as the anal pore. This is the anal pore. So the undigested food materials goes out through this. Same thing, ingestion, digestion, absorption, assimilation and egestion. Works. Now let's get into nutrition in human beings. I saw the time has already been passed by 1 hour, which is why I'm speeding things up. Okay. So this is my dear Papu. Okay. And this Papu will be doing nutrition. Will be showing a demo of nutrition. So over here, as you can see, starting from the mouth and going up to basically the anus over here, this is our gastrointestinal tract. Gastro as in the whole thing, gastro, stomach including esophagus, everything, intestinal as in intestine, and the tract, or it is also known as alimentary canal in simple terms. Okay, or is it also known as alimentary canal? These are the things. And over here, mouth, esophagus, stomach, large intestine and small intestine, everything becomes the part. Liver and pancreas are digestive glands. Digestive glands which gives out their secretion as well. Okay, going ahead, what do we have? Let's see. Parts of the digestive system. First thing, okay, we are going to go one by one. What happens in mouth? Mouth is the place where the first breakdown of food takes place, which is very much important. I have given 3 stars there. So mouth is a place where the breakdown of food happens. Now mouth obviously produces saliva. Guys, I'm going to be fast, okay? Saliva has 3 things. Bicarbonate ions that provides a neutral medium. So inside the food can be anything. It can be acidic also. Okay, a bit acidic. But to make things basic, neutral, okay, to provide a neutral medium, we have bicarbonate ions. It's not exactly neutral. Yeah, okay, fine. It's basically alkaline medium. Anyways, then the saliva also have lysozyme, which could kill the enzymes. You might have seen people say that you put saliva on the boys and things like that so that it get cured. Why? Because it kills all the pathogens. But your mouth has to be very clean for that. Next thing, salivary amylase, which is the star of the show over here. So saliva has 3 things: bicarbonate ions, lysozyme and salivary amylase. Salivary amylase is the enzyme which is responsible for the first breakdown of food in the mouth. Salivary amylase is also known as ptyalin, children. What is it known as? Ptyalin. P is silent over there. It is known as ptyalin. What does salivary amylase do? It converts. It breaks down starch, that is complex food, right, which we get from the whatever things that we eat. This starch into maltose, which is a simple sugar. Okay, maltose, which is a simple sugar. This is a complex sugar. This is a simple sugar. This is made up of so many glucose molecules. Over here, they are just made up of 2. So that's the difference. Now over here, you have to remember this also, that. It's happening in a near neutral medium, that is 6.8. Who provides it? Bicarbonate ions. So these are their friends. What is the name of the enzyme? Salivary amylase. It is also known as tyrin. What does it do? 30% of starch is getting broken down in the mouth itself, and it is breaking down into what? Maltose. Rest 70% happens elsewhere. I'll tell you about that. Remember, 30% is happening here, 70% is happening somewhere else. Okay. Going ahead, what do we have over here? Yes, if you talk about the mouth, this region, when we open up, this is the buccal cavity or the buccal region. Okay. Now the buccal region, when it goes backwards, this area, which is a junction between your nasal passage, your buccal passage, and also your esophagus and your windpipe, basically this junction is known as pharynx. So your food passes through the pharynx and then goes to the food pipe, and such that the food does not get entry into the windpipe. The windpipe is protected by something that is known as epiglottis, which is a flap of tissue. Okay, so food does not enter over there. Fine. Now one more thing, your teeth. Your teeth dentition, basically there is thecodont dentition, diphodont, heterodont. Heterodont means you have different type of teeth, canine, molar, premolar, everyone looks different. Diphyodont means you have one milk teeth when you chew. Then when you grow up, you have a permanent teeth, which is why diphyodont, as in 2 sets of teeth come in your whole cycle. Thecodont means your teeth is embedded in the jaw. Not everyone has that. Okay, that is what you mean by thecodont. This is something extra. If you want to remember, you can remember, but it will be really good if you remember. Okay. Now going ahead, what do we have? Esophagus. Okay, so after mouth, okay, after mouth we have something that is known as esophagus. That is the food pipe. Now, who is responsible for letting the things go into the food pipe and then move to the stomach? It is peristalsis. What do you mean by peristalsis? It is the involuntary contraction and relaxation of something. So over here, esophagus is undergoing peristalsis. It is not under our control. Our brain does it. It contracts, relaxes, relaxes all the smooth muscles over there, so that it moves, the food moves down to where, children? Down to the stomach. Now, inside the stomach, if you see the inner lining, suppose I am inside the stomach and I am saying, oh, there are pits over here. What are these pits? If you zoom this in, these pits are known as gastric pits. Okay, stomach, that's why gastric. So gastric pit is there, and inside each pit there are different type of cells, which I made in white, green and red color. The white ones are known as oxyntic or parietal cells. The white ones are known as oxyntic or parietal cells, and what do? They release HCl. They release HCl. So in your book, oxyntic and parietal is not mentioned, but if you can remember it, or if you do not want to remember, then say one type of cell is responsible for giving out what? HCl. But I will recommend you to learn it. Okay. Now, why do we need HCl inside our stomach? First thing, to kill the germs that comes along with the food. Second thing, to provide an acidic medium, because there are certain enzymes that are going to be released in the stomach itself, and they say we will only work if there is an acidic medium, or else we won't be working. So that is the scenario. So HCl is responsible for 2 things. It's very much important. First thing, I'm telling you again, it kills germs. Second thing, it provides an acidic medium for the enzymes to get activated. Okay, this is the first type of cell, oxyntic or parietal cell. Now going ahead, you have goblet cell, magic. So M for magic and M for mucus. So goblet cells are releasing mucus. Why? Because HCl is being released in the stomach. So obviously everything will get burned off. You don't want that. So you have mucus that protects the lining of the stomach from any damage from the HCl. It's something that you do not have to mug up. This is something understandable, right? Next thing, the third and very important, chief. Why do you call it chief cell? Because they are responsible for giving out enzymes. Okay, so over here I have just mentioned one enzyme. There are 2 more, but that is not of importance as of now. So it's for your midterm. So let's see. So there is an enzyme which is known as pepsinogen. So whenever no gen term comes, just understand that they are inactive. They are not active, they are inactive, children. So pepsinogen is an inactive enzyme. Now it converts into something active that is known as pepsin, and it occurs in an acidic medium. Okay, it needs an acidic medium, and who provides this medium? HCl, which is why it was very much important. If this medium is not created, this conversion won't happen, and if the enzyme is not active, then it won't work, obviously. So pepsinogen is the inactive form. If it is already active, what will happen? It will damage all the proteins up here, because this is a proteolytic enzyme. Proteolytic as in it is going to break down proteins. Okay, proteo as in protein, lytic, as in it is going to break down that. So it is made to be inactive because we do not want that to happen. We want it to be active only when it is needed, when the food is there. So during this point of time, acidic medium is given. 1.8 is the pH of our stomach, just imagine. Now, when pepsin is active, it breaks down proteins into something that is known as what? Peptides. So proteins are made up of peptides. Okay, so basically proteins are made up of amino acids. Few amino acids coming together, you call them peptides. Lot of them coming together, you call them proteins. So proteins are broken down to peptides over here. Peptides will again get broken down to individual amino acids also later on, not as of now. Okay, so this is the first breakdown of protein that happens, and it happens in the stomach. Okay, so stomach has 3 cells. First thing, they have gastric pits. They have 3-cell oxyntic parietal which releases HCl. HCl provides an acidic medium for the conversion of inactive enzymes to active enzymes, and also it kills the germs. Second thing, there is goblet cell which is responsible for releasing mucus, and this mucus protects the whole stomach's lining from the HCl. Now, who needs the HCl actually? Acidic medium is needed by the chief cells, okay, needed by the pepsinogen which is released by the chief cells. These are inactive. To make it active, you need an acidic medium that is 1.8. Pepsin is active. Pepsin works on proteins and breaks it down into what? Peptides. Okay, first breakdown of proteins happen over here. Going ahead, and remember, peptides also have to be broken down. It happens in the small intestine. So everything comes to its base model, okay, the simplest form in the small intestine only, because that is the last stop for digestion to happen, for the breakdown of things to happen. Remember that as well. So now, going it over here, we have liver. That is the next thing. So these are the digestive glands, as I said. What is the role of liver and pancreas? So this is the stomach that is there. Pancreas is over here. Your liver is over there. Liver secretes something which is very much important. It is known as bile juice, children. Okay, but liver secretes it, and it is getting stored in somewhere else. What is that somewhere else? It is gallbladder. So liver is the place where secretion of bile juice happens, and where is it stored, children? In the gallbladder. Okay. Now, bile juice is made up of bile salts and bile pigments, and a lot of other things, cholesterol, things like that. So bile salt is there, and bile pigment is there. Bile pigment as in bilirubin, biliverdin, which makes your susu yellow in color. Okay, so these are bile pigments are being made when hemoglobin breaks down. So there are a lot of things apart from that. As of now, let's not talk about that. Just remember, liver releases bile juice. Bile juice is made up of bile salt and bile pigment. What does this bile salt do? Okay, first thing, it makes the food alkaline. So whatever food is going to come from stomach, it is what? It is what, children? Acidic. Okay, so to make things alkaline, what do we have? We have the bile juice. Apart from that, it is responsible for emulsification of fat. It is very much important. What do you mean by emulsification of fat? Breakdown of larger fat globules to smaller fat globules. Okay, breakdown of large fat globules to smaller. So if there is one big fat globule, it is broken down to smaller ones. Why? So that the next enzyme which could come in, it could easily act on it. You tell me, surface area will be more for this or this? It will be more for this, because from all angles it can attack, and it can be very efficient, right? It will be so time consuming. This is so easy. So basically, emulsification of fat, that is just breakdown of fat. And remember, this is physical breakdown of fat. There is no chemicals being involved over here. This physical breakdown of fat globules into smaller compounds, so that the next enzymes could act on it. Okay, what are the next enzymes? We will see it. So as of now, liver, bile juice. Where is it stored? Gallbladder. Bile juice is made up of bile salt and bile pigments. Bile salt is responsible for making the food alkaline. Okay, because everything that comes from the stomach will be very much acidic now. And emulsification of fat, that is breakdown of large fat globules into smaller fat globules. Now, why is it happening? Because pancreas, our heterocrine gland, I'll tell about that. Pancreas is going to release pancreatic lipase. So whenever the term lipase comes, just understand it's for fat. It's going to act on fat. So now pancreatic lipase will be acting on the smaller fat globules and breaking them down into fatty acid and glycerol. Remember one thing, carbohydrates, when they are getting broken down, at the end, the most simplest form will be glucose. Okay, as I said, proteins, when they are broken down, the most simplest form will be amino acid. Okay, fats, when they are broken down, they will be forming individual fatty acids. There will be so many, 3 fatty acids will be there. Now there will be individual of them and glycerol. Okay, so in between this lots of things comes, which is written over here. But the final breakdown has to happen in the small intestine, please remember that. So smaller fats is broken down to fatty acid and glycerol, but these fatty acids are still attached to each other. They also have to get broken down. Okay, that will happen later on. Will you be able to remember this? Will you be able to remember this? Fatty acid and glycerol. Okay, pancreatic lipase is acting on smaller fats to break them down to this. Now pancreatic amylase is also there. Okay, pancreatic amylase is also there, and what is it acting on? Starch. Amylase also always acts on starch. Salivary amylase in the mouth and pancreatic amylase over here in the pancreas. So basically, this particular pancreatic amylase is going to act on the 70% of the starch that is still left to be broken down. Remember, I said 30% of starch is broken down there, 70% is still left, it gets broken down over here. Next thing, pancreatic trypsin. You have what? Pancreatic trypsin. What does trypsin do? Trypsin, pepsin, everything breaks down proteins. So whatever proteins are there, it gets broken down to peptides. Now you might be thinking, if these things are breaking down fats, carbs and proteins, is the food actually going to the pancreas? No. Pancreas, okay, so liver gives out its secretion, that is bile juice, so that when it comes and mixes over here, so all of them, they have a common duct which comes and opens up here. It provides an alkaline medium so that the pancreatic enzyme, whatever is being released, it can easily act on the food which is going to come from the stomach. So it's not like food gets in there, the pancreatic enzymes comes and acts over here. Okay, and this initial part is known as the duodenum or small intestine. Duodenum. This is the first part of small intestine where it is going to act. Okay. Will you be able to remember 3 enzymes? Pancreatic lipase acting on fats, breaking down into fatty acid and glycerol. Pancreatic amylase breaking down the rest of starch into maltose. And then pancreatic trypsin, which breaks down proteins into peptides. Once more over here, proteins are getting broken down in the stomach. In the stomach, pepsin was doing that. Here, trypsin is doing it. So remember, trypsin and pepsin is for proteins. Okay? Amylase is for carbs. Lipase is always for fat. Okay, now one more thing, pancreas are known as heterocrine gland. Why? Hetero as in 2 things might be there. That's why heterocrine. So basically, pancreas releases hormones, insulin and everything is released by pancreas itself. They are also releasing enzymes, that are these. So whenever a gland is responsible for releasing enzymes, you call it exocrine glands. Okay, you call them exocrine glands. Whenever a gland is releasing hormones, you call them endocrine glands. You will get to know about this when we talk about control and coordination as well. So since it is acting as both, you call them heterocrine gland. So it is a piece of information which is very much important for you. What is that heterocrine gland which is responsible for secretion of lipase? So you should be knowing it is pancreatic lipase which is being released by the pancreas. Okay. Going to the next stop, that is our small intestine. Small intestine, as you see over here, 3 parts are there: duodenum, jejunum and ileum. Ileum is the longest among them. Now if you see the internal structure, it looks like this. There is wavy things, and above that also there is wavy things, and above that there is something which is not visible over here. So I thought I'll zoom it up. So it looks like this. Now when I zoom the green part in, the green part also has certain things like this. What are these things? So basically, there are folds in your intestinal walls. On these folds, there are other folds which you call as finger-like projections, which you call as villi. And the villi over here, you might be seeing they are surrounded by certain cells which are known as, I think they are columnar cells only. They are columnar cells if I am not wrong, but I am not sure. Okay. Anyways, there are certain cells which have hair-like structures which are known as what? Microvilli. Okay. What are those, children? They are microvilli. Yes, it is ciliated columnar cells. Okay. So basically over here you have villi, and over that you have certain cells. These cells at the top of them, they have their cytoplasmic projections like this, which you call, for example, I will just zoom this and make once. It looks like this. So the cytoplasm itself is projected like that, and this thing that you see over here, this finger-like thing, extra thing that you see over here is microvilli. So the actual finger-like projection is your villi, and then above that you have cells like them, and at the top, these structures you call at microvilli. Why are you doing this? You want to increase the surface area for absorption. Now you only think if something is like this, it can absorb things, this much things. But if the same thing is made to be like this, then in the same space, two of them can be accommodated and more amount of surface area will be received for absorption of stuffs, which is why we have microvilli and villi. So in between each villi, you see there is a depth over here, there is an intestinal pit in between. So basically what happens, this intestinal pit will release enzymes. So the food that gets in over here, the enzymes will act on the food, okay, and whatever nutrients are there is taken up by the microvilli, basically the cells, and the cells gives it to what? Villi. The villi will be giving it to the blood vessels which are present over here. There is arteries and veins and also lymphatic blood vessels. There are also lymphatic blood vessels which you call as lacteal, okay. What does these lymphatic blood vessels do? So normal blood vessels, no, that is arteries and veins, they cannot take in fat. So these guys, the lymphatic capillaries, they take in fats as well. They have pores to take in fats as well, which is why it is very much important. So proteins and carbs can be taken, amino acids and glucose can be taken. But what about the other things? So for taking the fats, you have the lymphatic capillaries as well, which you call as lacteal. Now over here I have given you the intestinal pit. It contains the cells that releases intestinal juice. So there will be certain cells over here that will release intestinal juice or succus entericus. You call it as succus entericus. What does it contain anyways? It contains enzyme that does the breakdown of proteins, carbohydrates and fats. The final breakdown of proteins, carbohydrates into fats, that means carbs will be made into glucose, proteins into amino acids and fats into fatty acid, separate fatty acid and glycerol, okay. That is all about the small intestine, children. And herbivores, herbivores have a longer small intestine because they need to digest something that is known as cellulose, which is difficult to get digested, which is why they need a longer time to do so, okay. So please remember, this is an important assertion and reason thing. Herbivores have a longer small intestine because of the same reason. Now large intestine, if you see, there is colon, ascending descending colon is there, then rectum is there, then anus is there and anal sphincter is there. So anal sphincter muscle is something that controls the opening and closing of the anus. So for old people, know what happens, this sphincter muscle does not work very well because obviously their nerve connections and everything is loose now. It does not work as much as ours do. So they will be frequently doing their motion without their knowledge. So we should not be blaming them, we should be understanding their situation as well. Now large intestine is a place where digestion does not happen, okay, nothing gets broken down, only water reabsorption takes place. So lots and lots of water is getting reabsorbed by the large intestine, okay. That's all, children, that's all. Now one more thing, that is our teeth. So are we in this journey with me? Are you in this journey with me where we are learning a lot of things together? We'll complete it, okay. So what happens if you don't brush your teeth often, children? If you don't brush your teeth often, something like this could come, that is dental plaque. Let me tell you what the story about. So basically we have something that is known as enamel, okay, over our teeth, which protects our teeth from anything. Inside the enamel we have something that is known as dentine. Inside the dentine we have pulp cavity. Over here there will be blood vessels, nerves and everything. So slowly what happens, basically the bacteria, everything bores in, gets inside and causes a huge lot of trouble. But as of now, let's not go to that. As of now, let's see, you had some food, okay, you had some food, some food, whatever is it. Now bacteria will act on it. While acting on it, it will produce acids. This acid will go and act on enamel and cause trouble. This acid can go and act on enamel and cause trouble. So what is happening? It is acting on food, but when it's acting on food, it's like take the return gift. That is what? Acid itself. This acid goes and act on enamel and causes trouble. That is one problem. So food, if stays there, more acid is being produced. Now slowly dental plaque will be formed. What do you mean by dental plaque? There it is a mass of bacterial cell plus the food particles. So there is mass of bacterial cell and also the food particles. So because of this giant wall, saliva cannot act on the acid and hence neutralize it. So basically they are acting on the food particles, making more acid. The acid is going and making problem to the enamel. There's a huge lot of problem. But since there's a big wall being created, can our saliva go and act on the acid and make it neutralized? No, because of this particular wall which is being there, which is very thick, our saliva cannot go and act and say that enamel, I can save you. No, it cannot do that. Okay, because of the dental plaque which is being there. Now with this we are going to complete digestion as well. Big tap on your back if you have completed it with me. And again, question, most of the digestion and absorption of the food takes place in the, tell me, most of the digestion and absorption of the food takes place in the small intestine, liver, stomach or large intestine? It is the small intestine absorption. You see, you have microvilli, villi and everything. So small intestine. State the form in which the following are stored. Unused carbohydrates in plants. Tell me children, unused carbohydrates in plants, how they are stored? If you have got this correct, tell me, ma'am, I got this correct. Okay, that's starch. So you have glucose. This glucose is getting stored in the form of starch, which is unused, right? The energy derived from food in humans. So what kind of energy is being derived from food in humans? So when our food is getting broken down, I told you one thing is being formed, which is what? ATP. Even though this question suits well for respiration, we'll come to that as well. But yes, it is what? ATP. Adenosine. Adenosine triphosphate. Okay. Complete the flow chart. Gastric glands present on the wall of the stomach. This. So you have to tell the name of the cells, the secretion and their function. So first secretion will be what, children? It is let's say HCl. Okay. Second secretion is what, children? It is mucus. Third is what? Enzyme, that is pepsinogen. I am writing pepsinogen or basically pepsin, but you have to mention about pepsinogen. Okay, please mention pepsinogen, that it is getting converted to pepsin. Okay. What does HCl do? It kills the germs and provides an acidic medium. Kills the germ and provides acidic medium. Provides acidic medium. So I think I should not be wasting any bit of time by writing the whole answer, but yes. So mucus, it protects. Okay, mucus, it protects the lining. Stomach lining, and peptides, it's break down proteins to peptides. Pepsin. Done. Got it correct. Now let's go to respiration, children. After respiration we have circulation and then excretion. Okay, how is respiration different from breathing? This is a very important question. How is respiration or cellular respiration different from breathing? So breathing is a process where I am doing, this is breathing. Cellular respiration, I cannot see, it's happening inside the cells. Okay, so breathing is a very mechanical process which I am doing. Breathing is a mechanical action of getting in air in and out of the respiratory system. Inhalation and exhalation. Okay, now going ahead, how does respiration happen inside our body? So respiration happens inside all of our cells. Okay, it happens inside the cells. What is respiration? You have glucose, the food that you have. Okay, it is getting broken down to glucose. This glucose, which is a 6-carbon compound, it is getting broken down to pyruvate, which is a 3-carbon compound. This process is known as glycolysis, as glucose is getting broken down. Do not write glucolysis, please. It is glycolysis. Where does it happen? Inside the cell. It is happening in the cytoplasm. This is very much important. 10 times you read it, but please make it correct. Okay. Next thing. It's not just one, obviously, if it's 6 carbon. So when 6 breaks down into 2, which is 3 carbon, if this one thing is 3, so obviously other will also be 3. So there will be 2 pyruvate which is being formed with the help of one glucose molecule. Okay, and this process is still known as glycolysis. It is happening in the cytoplasm. Going ahead, all the rest of the process will be happening in the mitochondria. So, it was there in the cytoplasm, now it is moving into mitochondria, which is there in the cell itself. Okay, let's see what happens in there. Okay, first thing, just a second. Okay, this is perfect. So, if all things are fine, okay, if all things are fine, there is enough amount of oxygen, everything is there. So, basically at this point of time, what happens? Pyruvate gets inside the mitochondria, okay, gets into a cycle of reaction which you call as Krebs cycle. So, before getting into Krebs cycle, it is the pyruvate, it is getting converted to something that is known as acetyl-CoA, which is now going to enter the cycle, that is Krebs cycle. Why am I talking about Krebs cycle? Because Krebs cycle is the one that gives out CO2 and H2O. Okay. So, have you seen the process or the reaction, chemical reaction for respiration? C6H12O6 plus oxygen. So, oxygen is definitely required and it produces CO2, H2O and ATP. Major guy is this, but these things are also being produced over here. So, basically we saw how glucose is being utilized. We are going to see how oxygen is also going to be utilized. So, oxygen says I won't move out, I will be there in the mitochondria. So, oxygen is over here, which is why everything goes into the mitochondria itself. Okay. Now, as I said, pyruvate, it is getting converted to acetyl-CoA. This acetyl-CoA is going to get inside Krebs cycle, which is a cycle of reaction. During this cycle, no, CO2, H2O and lot of other energy molecules are being produced, GTP and NADP and things like that, lot of things. But what is the energy molecule that we need? ATP, because ATP has the highest amount of energy. So, what is the thing is that these smaller energy molecules, no, they get inside the mitochondria, they are already inside the mitochondria, but yes, they are being utilized, okay. All these things, GTP, NADP, everything is being utilized for the formation of ATP. And this process is requiring oxygen as well. So, it's a very big process which obviously we cannot learn today. It needs another 40 minutes of lecture. So, just understand what I am trying to tell you. There is glucose getting broken down to pyruvate, okay. Pyruvate gets inside the mitochondria, gets converted to acetyl-CoA, gets inside a cycle which is known as Krebs cycle. Now, in the Krebs cycle, CO2 and H2O is being released. Apart from that, there are certain other energy molecules which are being released, which will be helpful for the formation of our guy, that is ATP, okay. While this process is happening, oxygen is also required indirectly. Okay, oxygen is also required indirectly, which is why all these things are being produced. Now, since oxygen is being required, you call this process as aerobic respiration. You call this process as what? Aerobic respiration. Now, think of the other scenario where, for example, there is a cell, there is no enough amount of oxygen. For example, I started running, I ran, and now I am deficient of oxygen. I am tired as of now. At this point of time, my body still need energy to survive, right? It still has to produce energy. So, what does it do? This pyruvate, instead of going into mitochondria and doing all the stuff that is aerobic respiration, it gets converted to lactic acid and energy. So, energy is already being produced. That's the major thing. I have to produce energy, that is being produced, but along with it, there is a con as well, that is what? Lactic acid. Now, when this lactic acid is being accumulated in my muscle cells and things like that, it will cause what? Muscular cramps. It will cause cramps. But this, is it aerobic respiration? No, children. It is still respiration because energy is being produced, but this is an example of anaerobic respiration which is happening in the muscle cells when I am running and when I am deficient of oxygen, or in any part of a cell when it is deficient of oxygen. Okay. Now, there is another scenario. Suppose there is yeast and yeast is doing fermentation. At this point of time, again, there is no oxygen, nothing like that. You might have seen how wine and everything is made, right? Oxygen free containers are being used. Here also, basically, the pyruvate, okay, it is getting converted to one form of alcohol which is known as ethanol, and also carbon dioxide and also energy. Okay, ethanol, carbon dioxide, energy is formed, and this is fermentation. This is fermentation. This is another example of an anaerobic respiration. Here also oxygen is not being utilized. Okay. So in yeast fermentation, ethanol, carbon dioxide and energy is released. In our body, when oxygen is not there, lactic acid and energy is being released. In our body, when oxygen is there, carbon dioxide, water and energy is being released. And this whole process is known as respiration, cellular respiration, which happens inside each of a cell. Now this is entirely different from breathing, because in breathing you are taking in this oxygen which is required over here. If you do not breathe, no oxygen over here, no aerobic respiration. Okay. Now one more thing, the thing that we breathe out, carbon dioxide, where does it come from? From this same process. The cells, basically they are undergoing this set of reactions, which is why they are giving carbon dioxide, which is why I say carbon dioxide is a waste and is being given out. I hope you understand this particular thing. Okay. And one more thing I want to tell you. So you might be thinking, then why is actually oxygen required if energy is being produced? The energy being produced is very, very, very less compared to this. It might be 38 ATP molecules, it might be just 8 or 2. It's 2 actually. So that is the difference. So let's not take, and always remember, glycolysis happens in the cytoplasm, everything else happens in the mitochondria if it is aerobic respiration. If it is anaerobic respiration, it has nothing to do with mitochondria. Okay. Now there is a reaction over here which is given in your NCERT textbook and which is very much important. So yeah, one more thing. So when this process is happening, then also it is producing energy. Glucose to pyruvate conversion also gives out energy, which is being utilized for other purpose. So please do not forget this energy part. So there is a equation which I just solved, and then later my children told me, ma'am, there's energy as well. So that's how I remembered. So yes, there is energy as well. Please remember it. Okay. So there is glucose to pyruvate. It's known as glycolysis. It happens in the cytoplasm. This happens in the cytoplasm. Okay. This pyruvate can undergo three type of reactions. Absence of oxygen in yeast. If it's in yeast, it has to produce alcohol. What alcohol? Ethanol. Okay, and also carbon dioxide and also energy, which is why wine obviously we say it has a bit amount of alcohol. This is the amount. This is the thing that we're talking about. Okay. So ethanol, carbon dioxide and energy. Over here, in the case of muscle cells, it is lactic acid and energy. This lactic acid is the one responsible for cramps. Lactic acid is also having 3 carbon, just like pyruvate. Okay. Now, one that happens in the presence of oxygen, that is basically carbon dioxide plus water plus energy. Okay. That's all. So photosynthesis and respiration, you can do ulta. It's the same thing. So CO2 plus H2O is giving C6 H12O6 plus oxygen plus H2O plus energy, right? So if you do the ulta of it, do not put H2O over there in respiration. The same thing happens. C6 plus H12O6 plus O2 gives CO2 plus H2O plus energy. This time energy is on this particular side rather than that particular side. Okay. Going ahead, children, talking about plants. So how does gaseous exchange take place in the case of plants? So we know carbon dioxide they get from the atmosphere, and oxygen obviously. Oxygen also they get from the atmosphere. But in the process of photosynthesis, they take in carbon dioxide, they are giving out oxygen, right? How does diffusion take place? It takes place with the help of stomata. We learned about stomata. What are lenticels? Lenticels are found in woody stems, okay? So over here lenticels will be found. In the woody stems, lenticels are found. They do not have stomata. Instead, they have something that is known as lenticels, very much important. And also diffusion can happen through intercellular spaces. For example, these are cells on the outer layer of the leaf. There is space. Diffusion can happen from the spaces as well. Okay, so that is why it is intercellular spaces. So gaseous exchange in plants takes place through 3 different regions. One is stomata, second is lenticels, third one is intercellular spaces. Will you be able to remember it? It's very easy, right? So that's all. Now going ahead, how and what happens. So it's a daytime, even the black background is there, but it's daytime. So what is happening? Carbon dioxide is being taken up. Okay. Process happens. Oxygen is being released. That is also fine. But I should be using white. Okay. But. Now, if you are asked, is oxygen being released more or carbon dioxide being released more? So you might be thinking, how is carbon dioxide released? Carbon dioxide is utilized, right? Well, carbon dioxide is released during respiration, where oxygen is being taken in, and carbon dioxide is given out. So there should be release of carbon dioxide as well, right? But what happens, children, in daytime plants need carbon dioxide. So they say give the carbon dioxide back, do not give it outside. So carbon dioxide is not given outside, instead it is taken again for photosynthesis to happen, and hence the oxygen is the one thing that will be in more amount. Okay, didn't get it? Once more I can tell you, listen to me very carefully. So what happens? Usually in photosynthesis, carbon dioxide is taken in, oxygen is given out. Agree? But there is another process which is known as respiration, where oxygen is taken in and carbon dioxide is given out. But this carbon dioxide cannot be given out as a process for photosynthesis is taking place. So hence, carbon dioxide is utilized in the process of photosynthesis. There will be a little amount of release of oxygen during photosynthesis. So in daytime we say the major amount of gas which is being released, it is oxygen, since carbon dioxide is used in the process of photosynthesis, which is not entirely correct, but yes. Okay, so that is correct. What is not entirely correct is what is written in your NCERT. Your NCERT states that there is no CO2 being released during daytime. Okay, this is given by your NCERT. We have to follow your NCERT, but let me tell you, it's not like that. Little amount of CO2 is also being released, but we have to follow your NCERT. So let's follow. So in daytime, what is the oxygen? What is the gas that is being released more? It is oxygen. Okay, remember that. Now if we talk about night, what happens? Carbon dioxide, is it needed to be taken up for photosynthesis? Will photosynthesis take place in night? No. So instead what happens, children? Whatever oxygen is there in the atmosphere, it is being taken in for the process of respiration, and hence during the process of respiration CO2 is given out, and this CO2 will be the one that will be released outside. Now when CO2 is being released outside, there will be more amount of CO2 outside. Photosynthesis is obviously not happening, respiration is happening, so there is no need of CO2 in there. So at night time CO2 release is more and oxygen is being utilized for the process of respiration. Okay, clear with that? So there is a statement that is given in your NCERT. At night, when there is no photosynthesis occurring, CO2 elimination is the major exchange. Okay, CO2 elimination is the major exchange when, children, at night. During the day, CO2 generated during respiration is used up for photosynthesis. So whatever CO2 is being released during respiration is used for photosynthesis, hence there is no CO2 release. Instead, oxygen release is the major event at night. So at night, at this time, at this time. So yes, I'll just write it out for you. Morning, it's oxygen. Night, it's carbon dioxide released in more amount. Okay, your teacher is also getting tired. Okay, chalo. Gas is exchanged in fishes. That is kind of very different, something new that we have to learn. Chalo. So gaseous exchange occurs through gills, which takes in the oxygen dissolved in water. So they definitely have gills, and these gills are the ones that basically filters out oxygen, takes into the blood vessels. We are going to see how. But there's a very important question over here that is put down. Why do fishes breathe very fast? The answer to this is, I am tired of taking 100 breaths per minute. There is so less dissolved oxygen up here. That means the amount of oxygen which is there in the water bodies, it is very, very less. And this oxygen is not the gaseous oxygen that we have, it is in the form of dissolved oxygen. And the dissolved oxygen is less, children, in the water bodies, which is why it has to take so many breaths per minute, so that it can take more amount of oxygen. So it's taking in water, but little amount of oxygen is coming, there's no use, which is why it is breathing very fast. I hope you understand. Now over here, I have the breathing pattern in fish. Let's see it. So there is water. In the water, there is dissolved oxygen. The water moves inside the fish, moves outside to the gills. Okay? But whatever oxygen is there, it is being taken up by the blood vessels which are going to surround the gills. Now, the blood vessels have the oxygen, yeah, this oxygenated blood goes to the different body parts, wherever it is needed. Now the body parts takes in the oxygen, and what is the cellular waste, children? We saw in respiration, CO2 was the waste, right? So this CO2, okay, this CO2, that means the blood which will now return back will be having more amount of CO2 and very less amount of oxygen, right? This deoxygenated blood goes to the heart pumps the blood back to the gills, okay, into the vessels or the gills, and the same thing repeats. So here one very important thing, the heart in the case of fishes never receives oxygenated blood, they always receive deoxygenated blood, okay, that is a very sad truth. And over here I remembered one thing, in respiration, no children, in respiration these are the byproducts. These are the byproducts, okay. If they are asking about the byproducts, please remember CO2 and water is the byproduct with human respiration. So again, diagram is very much important, you have to study this, okay. So breathing in humans start with the nostrils or the nares. You have external nares, the same term comes, do not get worried. External nares, okay, then there is internal nares which takes in. Now the air moves around, okay, I am going to highlight things. Air goes in, oh, nothing is will be seen over here, right? Yes, nothing will be seen. So anyways, air moves over here, okay, and then should it go to the foot pipe? No, it goes to the windpipe. Over here epiglottis opens up, and there comes this particular region, okay. Epiglottis is the flap I said, right? So then comes this particular region. What is this particular region, children? It is the trachea. It is the trachea. So over here you can see, where is the trachea, children? Yeah, trachea is there, and trachea basically have rings of cartilage around it. You see this? There are rings of cartilage around it, so that the trachea does not collapse when air is not there. So this is a very thin structure, so to not make it collapse, what do we have? Rings of cartilage surrounding it, okay. Now trachea comes in, it breaks down into two, that is what? Bronchi. It is what? Bronchi. Now bronchi also will break down into further branches, they are known as bronchioles. Bronchioles will further open up into small air sacs which are known as alveolus. What is it, children? It is the alveolus, and everything of this is covered with the help of ribs, okay, which protects everything, your heart, everything. And the middle part of the middle part is known as sternum, okay. I am not writing it over here to not make you confused. Now what happens in the alveolus? Why is it very much important? Because alveolus is the place for the major exchange of gases. Please write it down somewhere. Sorry, don't write, just listen to me. Major exchange of gases takes place in the alveolus. So just imagine there is an alveolus, okay, there is blood vessel around it. Over here respiration is happening inside our body, which is why deoxygenated blood is coming, okay, which is having more amount of CO2. Now you only see oxygen is more here, oxygen is not here, it is comparatively less. So oxygen moves down over here and CO2 moves up, okay, because of the pressure difference that is being there. Now the second scenario will be, it will look like this. Now there will be oxygen and there will be CO2 here. It's not like continuation, it's the second scenario that I was trying to show, 1 and 2. Okay, now this oxygen will go to the heart, oxygenated blood will go to the heart, it gets pumped to different parts of our body and then comes back to the heart and then back to the lungs with the deoxygenated blood. So major exchange of gases takes place in the alveoli or the air sacs because they are very thin in structure, they are surrounded by blood vessels through which everything can get easily transported. Okay, I hope this is clear. Now over this you have to remember your breathing will be left incomplete if you are not talking about diaphragm. Now diaphragm is very much important. Over here you have a flap of muscle that is seen over here, and you have to remember what happens when diaphragm contracts and what happens when diaphragm relaxes. So basically you can think like this, even though it's a not very nice method of explaining. So when no one is there, when I am in a relaxed position, we all bend like this, right? We all bend like this. Similarly, okay, when diaphragm is there in relaxed position, it also gets curved like this. When it is curved like this, that means there is a very less amount of space in this particular region, which you call as what? Thoracic cavity. That means you are, as of now, you have breathed it out. Okay, you took you did exhalation, you breathe it out. Okay, yes. So basically it relaxes and it moves up. Okay, relaxes and it moves up. So whatever air is there goes out. So while breathing out, the diaphragm relaxes. And when what do you mean by relaxing? It means that you are bending down. So we get confused, even I got confused now also. Why? Because we think relaxing means it will now lie straight. It's not like that. Okay, when it lies straight, there will be too much amount of space. It's not happening like that. When you breathe out, that's when the diaphragm is relaxing. And when I say diaphragm is relaxing, that means it is bending. Okay. Now, on the other hand, when you talk about breathing in, you need more space in the thoracic cavity, where everything is present, right? So you need to contract the diaphragm. Now the diaphragm contracts. Okay. When the diaphragm contracts, when someone is there, you be like this, right? Same thing happens over here. It becomes all straight. Okay. Will you remember this? So during breathing out, it will be relaxed. That means it will be like this. Okay. Thoracic space will be less. Please remember. Now, one very important thing, if you want to remember the the order. So metabolic poison leaves the body by airways. What is metabolic poison? It's basically carbon dioxide. So M for mouth, P for pharynx, L for larynx, T for what is this? Trachea, B for bronchi, B for bronchioles, A for alveolus. You can remember like that as well. Okay, that's a perfect way. So I hope you have got that also. So can diffusion pressure alone help with transport of oxygen? Can diffusion alone help with transport of oxygen, children? Just for example, I have stopped breathing where there is no importance of lungs over here. There is air around and I want all the different parts of my body to get this particular oxygen rich air that is present over here. Will my body be able to get it? Answer is no. Why? Because diffusion is a very, very, very slow process if you take on large scale. So for smaller organism, it's possible. Okay, unicellular organism, it's possible. But for us, we are multicellular organism. We are more complex. No, because we are multicellular complex organisms who have high metabolic needs. You remember metabolism, right? That means so many chemical reactions are happening, which is why we need more amount of oxygen. Okay, so that is the reason why diffusion pressure is not alone. Diffusion will be so slow, we will die. We don't want that to happen. Okay, now two very important that is given over here in your NCERT textbook. Hemoglobin has higher affinity to oxygen than carbon dioxide, which is why hemoglobin will be really happy to carry oxygen. You can remember like that. Hemoglobin. Okay, hemoglobin has lots of O, so O for oxygen. Carbon dioxide is more soluble in water and hence can be transported in dissolved form. So in our body, along with the, okay, carbon dioxide is transported in the RBC also, outside the RBC in the plasma also, because the carbon dioxide can be dissolved. It is more easily soluble as it is written over here and can be transported in the dissolved form. So if the question comes, you need to answer it. Done? Okay. Okay. Chalo. One experiment to show that we are giving out carbon dioxide. So basically there is a pitchkari and this is lime water. Okay, what is this, children? This is lime water. So basically when you pump the pitchkari inside, it does not have carbon dioxide and things like that, which could convert this lime water milky. Okay, when carbon dioxide is there, carbon dioxide reacts with lime water, that is CaOH twice. When it reacts, it forms calcium carbonate. And this calcium carbonate is an insoluble, it forms milky white color, it's an insoluble precipitate. So I am writing calcium carbonate, okay, which forms the cloudy or milky white color. Whereas this is the lime water. Lime water is also known as CaOH twice. Tell me, any chemistry fans over here? Carbon hydroxide. Oh, sorry, calcium hydroxide, carbon hydroxide. Oh my god, the chemistry teacher who taught me might be in utter disappointment as of now. Let me make it calcium hydroxide. Okay, works. So when carbon dioxide is there, it is going to turn milky. As you can see over here, it is not being seen over here, but yes, when a person he breathes out, so it is giving out carbon dioxide, and this carbon dioxide will make things milky. Okay. That is the experiment. Okay. Diffusion is insufficient to meet the oxygen requirements of multicellular organisms like humans. State the reasons. I told you the reason. We are complex. Okay, we are complex multicellular organism. We have high energy needs. We have high metabolic needs, which is why diffusion is insufficient, because there are so many cells in our body through which the oxygen has to be diffused, and it's a very slow process that is going to take place there. 4 things you have got. Okay, slow process. Complex organism are there. Multicellular, we are multicellular. We have high metabolic needs and we have high energy needs as well. Okay, write it very properly. In the process of respiration, state the function of alveoli. You know, the major exchange of gaseous exchange takes place where? Alveoli. It is the air sac which is getting broken down, which is branching off from the bronchioles, right? And they are richly supplied with blood vessels which will be carrying oxygen, which will be carrying carbon dioxide rich blood. The carbon dioxide moves into the alveolus, then it goes to from the alveoli. This is the alveoli. From here it goes back to the bronchioles, bronchi, trachea, then outside. Okay. So you have to tell about all of that. Rate of breathing in aquatic organism is much more faster than in terrestrial organism. Give reasons. Why, children? Again, in aquatic organism, the what? Dissolved oxygen is comparatively very less. Okay, dissolved form oxygen is very less, which is why they have to breathe fast so that to get more amount of oxygen for respiration to take place. Complete the following pathway showing the breakdown of glucose. Chalo, let's solve. You tell me. Anyone in the comment section? Come on, come on. Glucose in the cytoplasm, it is getting broken down to 3 carbon molecule. What is that? And energy. What is this 3 carbon molecule? It is pyruvate, my dearest little children. Now, in the presence of oxygen in mitochondria, this pyruvate is again getting broken down to dash, dash and dash. So, dash H2O and energy. Carbon dioxide which we breathe out. Who all got it correct? You don't worry, you are going to get this thing, homework as well. Now again we are going to start transportation and we will be finishing transportation and excretion, all of them together. So I want you to take a break, take a quick recall, have enough amount of water and then come back. I hope you drank your water as well. Well, you should be hydrating yourself. I'm a biology teacher. I know how much important it is to do that, and also we are going to talk about excretion of water. So definitely let's see how the process works in real also. So let's go ahead with transportation, children. So transportation is a process where you are transporting important nutrients, you are giving out the cellular waste, you are providing a very right pathway for everything that is not useful for our body to go outside. Okay, so it is collecting waste as well as distributing what is important. Starting with the heart, our heart has 4 chambers. Okay, 2 atrium, 2 ventricle. Okay, this is white. We have 2 atrium and 2 ventricle, which is why we say we have 4 chambered heart. 1, 2, 3, 4. Okay. Now, basically you can remember like this. First thing, let's start with first only. In our body, we have so many roads. Okay, we have so many roads, and on these roads, which are blood vessels, which are what? Blood vessels. On these roads, there are so many vehicles. And these vehicles are our blood cells. But these vehicles need an engine, and that engine is our heart. If our heart is not working, obviously blood won't be moving around. Okay, so this is all about transportation, and now let's begin. So the heart, when we talk about the engine which is going to pump the blood, it has 4 chambers, human heart, 2 atrium, 2 ventricle. Okay, one side is right side, the other one is left, which is why I wrote RA, right atrium, right ventricle. Over there, left atrium, left ventricle. So there are so many arrows and everything given over here. Just concentrate on what I am trying to tell you. Okay, now you know, in the lungs, the alveolus has just given the blood vessels oxygen. Now blood will be coming from the lungs via the blood vessels, and now it has to get to one part of the heart. What is that part of the heart? It is the left atrium. And how can you remember it? You can remember LAO, that is bring in oxygen. I use this trick everywhere, so please forgive me for that. But yes, LAO as in bring in oxygen, or LA. Oh, Los Angeles. You can remember like that also, either way. So it has come now. The oxygenated blood, rather than going to the right side, it has come to the left side. Please remember, oxygenated blood comes to the left side, La O. Okay. A comes first, then comes V. So atrium comes first. So atrium receives it. It goes to the ventricle. Now all the cells of our body are saying, give us oxygen, we need to do respiration. So basically from LV, that is left ventricle, the blood is going to go to different body parts. And this blood is oxygenated for obvious reasons, because it is coming from the lungs. Now all the cells becomes very happy, like we got oxygen, but we have to give you something back, that is CO2. So blood cells, it gives back the blood vessels CO2, or the blood cells, the body parts, it gives the blood cells CO2, and this CO2 rich blood, that is deoxygenated blood, will now again go to the left side. No, it goes to the right side this time, because you know LaO is there. No, oxygenated blood is over there. Deoxygenated blood will be over here. Okay. So this deoxygenated blood is going to go to A first and then V. So it goes to A and then goes to V. Okay. Now when it reaches the ventricle, it is now going to pump the blood to the lungs. What type of blood, children? Everything is deoxygenated over here. Deoxygenated blood will be going to the lungs. Agree? Do you agree on that? And now the same thing happens. Alveolus gets enough amount of oxygen when we breathe, and then it goes to the blood, and the same thing repeats. So as of now, you can just imagine RA, RV, LA, LV, lungs receive oxygenated blood. It goes to LA because LA is asking La O. From here it goes to LV. From LV it goes to different body parts. The body parts, obviously they are doing respiration. So CO2, cellular waste is there. Cellular waste, that is CO2 is there. This CO2 is now taken back to RA because A comes before V, then V, from V directly to the lungs, and the same thing repeats. Okay. Now let's see who is responsible for what. So whenever lungs is there, just imagine the word term pulmonary will be there. So pulmonary is here as well. Now there's a usual notion that arteries are responsible for carrying oxygenated blood always. But yes, it is true in some sense, but there is an exception as well. What is an exception? Over here. Over here you see artery, pulmonary artery is the one that carries blood from the RV to the lungs. Pulmonary artery is the one that carries blood from the RV to the lungs. Pulmonary vein is usually related to deoxygenated blood, but here again it's an exception. Pulmonary vein is the one that carries oxygenated blood from the lungs to the heart. So please remember, basically over here pulmonary, whenever the term pulmonary, they are the exceptions and they are doing the opposite of thing that they were usually doing. So pulmonary vein is going to carry oxygenated blood, pulmonary artery is going to carry deoxygenated blood. Now the one artery that carries blood to different parts of the body, the biggest artery is aorta. What do you call? Aorta. Okay. And then comes the one that carries back things, that is known as vena cava. What do you call it? Vena cava, there are two, superior and inferior. Okay, superior ones collect the blood from different upper parts of the body, whereas inferior one collects from the lower part of the body. Okay, so you have superior and inferior vena cava over there as well. So total 4 blood vessels for you to remember. Pulmonary artery, okay, pulmonary vein. Pulmonary vein is an exception that carries oxygenated blood. Pulmonary artery is an exception that carries deoxygenated blood. Okay, now over here you have an artery which is the biggest, largest artery. Okay, it is aorta. Over here you have vena cava. So on the other side you have one vein and one artery, if you see, and this is vena cava. That's all. Going ahead, we have valves as well. So basically what happens, children, the blood which is coming over here, it is getting pumped down. So when it is getting pumped down, this particular atrium gets contracted, right? So this guy comes down. But now ventricle, when it contracts to send things to the lungs, don't you think the blood could move back to the atrium? Yes. So to prevent that, you have two walls. One over here on the right side, you call it tricuspid. So you can remember ri for right side, okay? So tricuspid as in there are three valves like this, which is why it is known as tricuspid. Over here, there is bicuspid. Okay, they just have two. So tri is there and bicuspid is there. Bicuspid is also known as mitral valve, and they both of them are atrioventricular valves. What do you mean by that? Atrioventricular, it's simple. The term is very complicated for you. It's very simple. Atrium, ventricle. Between that, the valves which are there, they are known as atrioventricular valves. Okay, one is tricuspid and the other one is bicuspid or mitral. Done. There is one more valve over here also. When blood is being pumped down, okay, there are high chances during some time the blood could come back again from the aorta. So you have aortic valve and also pulmonary valve. This is easy, right? Aorta, so aortic valve. Pulmonary artery, so pulmonary valve. That's all. They are semi-lunar valves. Why do you call it semi-lunar? Because it is like half moon shaped valves. That is why it is known as semi-lunar. Lunar as in moon. Okay. So we studied there are 4 chambers. Okay. 2 atrium, 2 ventricle. Okay. Deoxygenated blood comes to the left side of the heart. Okay. And the one that carries the blood towards the heart is the pulmonary vein. The one that carries away from the heart to the body parts is the is the aorta. Is what? The aorta. Now one that carries blood towards the heart is again superior and inferior vena cava, and then the deoxygenated blood which is transported from the heart to the lungs, it is what? Pulmonary artery. One very important thing to remember, okay. A for away from heart. So arteries always carry blood away from heart. So if you're finding it difficult to remember the names, okay, just see blood is coming over here, no? It's coming towards, so it has to be a vein. It is going away, so it has to be an artery. Again vein, again artery. Okay? Vein is towards. Okay. Now, okay, valves are also there. Tricuspid is there between this one, and bicuspid is there over there. Aortic and pulmonary valve. Going ahead, we have 2 types of circulation which is known as double circulation. We say we human beings have double circulation. It's a very important topic. Listen to me very carefully. Okay. So what happens? Pulmonary circulation is when blood moves between the heart and the lungs. So over here blood is moving from the heart to the lungs. Again deoxygenated blood comes, and again the blood is moving from the heart to the lungs. This is pulmonary circulation, where once oxygenated blood is coming and the once deoxygenated blood goes. Okay, this is just between the heart and the lungs. Okay, that is why pulmonary circulation is there. Next thing, there is systemic circulation, where the blood moves between the heart and the body. So oxygenated blood goes, deoxygenated blood comes again. So this is between the heart and the body. This is system, body system. So that is what you mean by double circulation. So once I am trying to tell you that in a whole cycle, the blood is traveling twice through the heart. Okay, it is traveling twice through the heart. If you see, once was this, second one was this. Similarly, once was this, second one was this. So that is what you mean by double circulation. Okay, and in double circulation, you have 2 things, pulmonary circulation to remember and systemic circulation to remember. Now, one thing, wait, yeah. What is the difference over here? Let's see. This is an important question, so you can take it in the format of question as well. Pulmonary circulation, this circuit involves the flow of blood between the heart and the lungs. There it is, heart and the rest of the body. Oxygenated blood from the lungs open into the left. You can explain how oxygenated blood comes to the lungs and how then deoxygenated blood from the right ventricle is carried back to the lungs. Everything that comes in between heart and lungs you can explain in detail. Now, very important thing is that we humans definitely show complete double circulation. We have double circulation, but it's not the same with other organisms. So we are going to talk about that. So for that I have to talk about this first. Let's see. Who has the superpower of maintaining a constant temperature? So among organisms, if you see over here, there are homeotherms and poikilotherms. Do you know about them? Do you know who are homeotherms and who are poikilotherms? Homeotherms are also known as warm-blooded and poikilotherms are known as cold-blooded. So who has the superpower of maintaining a constant temperature? Children, it is homeotherms. We are homeotherms. Okay, we are homeotherms. We can maintain a constant temperature. Whatever temperature is there outside, our body will be having 37 degrees Celsius. That's the end of it. But poikilotherms are not like that. They cannot maintain a constant temperature, which is why they will be like, for example, it's too much hot outside, then they will go near water bodies or stuff like that. Okay. Or there's very much cold outside, so they will go be in some sunny area, things like that. So things works like that for them. They do not maintain a constant body temperature. They do not have that much energy. Now this is the very important thing. What is the difference between homeotherms and poikilotherms? How they are able to maintain a constant body temperature? It's not a free process. For this, they are spending out lot of money, and this money is in the form of oxygen. Do you think without oxygen energy can be produced? No. So basically oxygen helps in the production of energy with the help of a process which is known as respiration. Agree? This energy is being utilized to maintain a constant temperature, which is why we need a clear-cut differentiation between oxygenated and deoxygenated blood. We cannot afford our blood to mix. So there are organisms in which the blood mixes. So we cannot afford that. Now if we talk about fishes, they do not have enough oxygen at all. That's why they breathe fast. So let's keep them aside. There are other organisms like frogs. They are known as amphibians. Why they are amphibians? They live on land, and that is different, but they have a very common area where the deoxygenated blood and oxygenated blood get mixed, because they do not have to maintain a constant body temperature. So I'll tell you who are the ones that are homeotherms. Human beings, or basically mammals, are homeotherms, and then aves. Mammals are homeotherms and aves. Aves means birds. So birds and us need a constant body temperature which is being maintained with the help of respiration, which provides enough amount of energy. But when you talk about poikilotherms or cold-blooded, there is 3 of them. Pisces, that is fishes. Okay, the name Pisces will come, do not forget. Pisces, amphibians, reptiles first, then amphibians. Reptiles as in your snake and all, okay? And amphibians as in what? Your salamander, frogs and all. They do not maintain a constant body temperature, which is why they are okay with mixing of blood. Okay, they are okay. These guys are okay with mixing of blood. This guy is not getting enough amount of oxygen only. So how can it maintain a constant body temperature? Okay, so I say that we show complete double circulation. Okay, we have a proper set of guidelines which is there in our body. No mixing of blood. Okay, which is why we have 2 circulation, pulmonary and systemic. Both aves and us. But over here, it's not the case. They have mixing of blood, reptiles and amphibians, and Pisces do not. We will see about them as well. I hope this is clear. Oh, they are known as poikilotherms. We are homeotherms or warm-blooded. They are cold-blooded. Okay. Now let's see fish's heart. We already studied about fish's heart, so I'm just going it very fast. Okay, listen. So gills have blood vessels from where the oxygenated blood will come. So I'm writing oxygenated blood will come. It gives to the rest of the body, and now carbon dioxide rich blood, deoxygenated blood goes back to the atrium, to the ventricle, and from the ventricle back to the gills. So this time over here first thing, they have just 2 chambers. That is the first difference between us and them. That is the fishes. They have just 2 chambers, we have 4. And they never receive oxygenated blood. No oxygenated blood ever. Please remember that. Okay, they always get deoxygenated blood. Going ahead with the heart of reptiles and amphibians, they have right atrium, left atrium, and just 1 ventricle. This is not right ventricle, this is just 1 ventricle. So lungs give them oxygenated blood. This goes up here in the ventricle. Okay, from the ventricle it goes to the rest of the body. From the rest of the body, deoxygenated blood comes to the right atrium. This deoxygenated blood goes to the ventricle. So basically over here you will be finding mixed blood. And are they okay with mixed blood? Yes, because they do not have to expend too much amount of oxygen for formation of energy. Okay, but there are exceptions as well. So in reptiles and amphibians, you know, there's crocodile. Crocodile is a reptile and it's different as well. It has 4 chambers. It has to maintain. Its lifestyle is different, which is why it has 4 chambers of heart. Please remember, any organism that belongs to crocodile family, alligator, gharial, whatever is there, all of them, they have 4 chambers, and it's a reptile, it's an exception, so you have to remember it. Okay, and they have mixed blood and they show incomplete double circulation, because you know Incomplete double circulation, there are proper segregation between heart and lungs and everything. Over here they are showing incomplete double circulation, but yes, they show double circulation, okay. So whenever you write this thing, please understand that for double circulation there is what? For double circulation there is human beings, or basically mammals, and what children? Mammals, aves, reptiles and amphibians, all of them, they show what? Double circulation, okay. Remember that. These ones are doing incomplete, but they're still doing. And over here the blood is passing just once through the entire cycle, which is why fishes, they have single circulation. Fishes are the only ones that have what? Single circulation. Going ahead. Okay. Why mammals and aves show double circulation? Important for both. So mammals and aves are warm blooded, which means they need to maintain a constant body temperature. Now efficiently separates, if there is a double circulation, it separates oxygenated and deoxygenated blood. This efficient separation helps in providing oxygen needed for ATP formation. I have written all the answer, please read it out once, okay. Going ahead, okay, now let's talk about the blood vessels. We have 3 in number majorly. One is artery, the other one is capillaries, and then is veins, okay. The arteries among them, okay, arteries, this is the structure. So arteries basically, they break down into arterioles, they break down into capillaries, similarly veins, venules, and then capillaries. So I hope you remember this. Going ahead with the difference, it will help you to understand things much better. So arteries, as I said, they are taking the blood away from the heart, Veins, they take the blood towards the heart, okay, and capillaries is where the maximum exchange of gases takes place. So you might have heard about this in alveoli. Same thing, capillaries are very thin, okay, they are very thin, we'll talk about that, which is why maximum exchange takes place over here. I'm not writing anything. Okay, now pressure, if you see arteries, since heart is directly pumping the blood, it is taking the blood away from the heart, right? So the pressure that the artery has to suffer is way too much. Okay, so there will be high pressure. What about veins? They are taking blood from all the different parts of the body to the heart. There is no pressure, so low pressure. Similarly, capillaries also low pressure. If we talk about the diameter, arteries are narrow. Why they are narrow? I'll tell you. Why they are narrow? Because they have 3 layers, and 3 layers. If you see, the muscular layer is very much thick. All the layers that is there, it is very much thick because they have to maintain or suffer through this much pressure. If they are not thick, probably our blood would get leaked, our blood cells would get damaged, our blood vessel would get damaged, which is why they have very narrow lumen. There is only narrow space because they have very much thickened wall. Okay, they have thick wall and hence narrow space over there. Over here, in the case of vein, they have thin wall. They have all these layers, but it is very thin. Now, since they are thin, obviously it will be, the inner area will be wide, it can carry more blood up. Now, if you talk about capillaries, they just have 1 layer among them, that is tunica intima. You do not have to learn this. You learn this, just understand, if this is a capillary, it is just made up of 1 cell thick membrane, that's all. Just 1 cell thick layer is there over here, that's all, nothing else. So it is extremely thin over there. Now, muscle and elastic layers, as I said, is present in large amount, which is why it is thick, small. Now, valves, that is very much important. Valves, why do we need valves? So, your heart is pumping blood towards your body, so there are no chance that it will give the blood back. Very less chance, very less, nil, almost nil, because it is under high pressure. At this point of time, you do not need valves to prevent the I'll make it in white. You do not need valves because blood won't be going back. But in the case of veins, it is carrying the blood from the lower regions. So obviously there are chances that the blood will move back, which is why they have valves. So remember, V for veins and V for valves. Okay. Capillaries also do not have this. They are very small structures. So what do you expect from them? And capillaries have small pores as well in between. Okay. So I hope this is also done. We do not have time to revise. We have to go ahead. Now, if we talk about blood pressure, what is there about blood pressure? What do you mean by blood pressure? So the force that the blood exerts against the wall of a vessel is known as blood pressure. So you have your heart, the ventricle is there, which is giving the blood to the aorta. Now when it does, okay, basically when the blood comes, it is going to put some pressure on the walls, right, of the blood vessel. This pressure that the blood is putting, it is known as the blood pressure. And if it is low or high, you will get to know that your heart is not working properly. So what is it? The force that the blood exerts against the wall of a vessel is known as blood pressure. There are two, okay, systolic and diastolic. What do you mean by systolic? The pressure of the blood inside the artery during ventricular systole. What do you mean by ventricular systole, children? So just imagine there is, okay, so I am explaining everything over here. So this is the atrium and this is the ventricle, okay. Now what happens? Ventricle, when it contracts, there will be too much amount of blood that goes out. At this point of time, the pressure is really high because it is contracting, right? Ventricular systole, the term systole means contraction. The term systole means contraction. So when ventricles are contracting, the pressure on the walls of the blood vessels are going to be high. So it is 120 mm of Hg, of mercury. That is the systolic pressure, normal systolic pressure, 120 mm of Hg. Going ahead, talking about diastolic pressure, it's when this particular ventricle is now going to relax. When it now relaxes, what will happen, children? Obviously the pressure becomes less, okay. It is known as diastole, ventricular diastole. Diastole means relaxation. So during ventricular diastole, what is happening, children? The pressure is less and it is 80 mm of Hg. So what is a normal blood pressure? 120 by 80 mm of Hg is the normal pressure. Systolic is the contraction, ventricular contraction, and diastolic means ventricular relaxation. I hope you got it. Now, what is the instrument used to check the blood pressure? It is sphygmomanometer. What is it? Sphygmomanometer. Sphygmomanometer. Tell it 10 times, you will remember it. But you have to remember it. Questions have come again. Okay. Going ahead. So, blood vessel is done. Okay. Now, we have blood cells. So, blood cells, when you talk about them, there are erythrocytes, that is the RBC. Okay, which is responsible for transporting oxygen, and because they have hemoglobin, they transport oxygen and give out carbon dioxide. They take in carbon dioxide. Next thing that you have is white blood cells. White blood cells which are going to fight against diseases. So white blood cells, basically leukocytes, there are different types of them. There is lymphocyte, monocyte, eosinophil, neutrophil, basophil, all of them having different function. Okay. Basophil is responsible for inflammation. When inflammation will be there, a lot of other cells could come in and help in that particular region, which is why inflammation is very much important. Neutrophil is responsible for taking in whatever pathogen comes in inside its body. It is known as phagocytosis. Same with monocyte, it is also responsible for the same thing. Eosinophil is responsible for allergic responses. If your body is not showing allergy, then it will be a problem. Your body is not able to take it, which is why allergic responses comes in. Lymphocytes are the one that produces antibodies, okay, when something comes in, or kills the virus cells and lots of things like that. So your leukocytes or WBCs are responsible for fighting against diseases. Okay. Now if you're talking about platelets, what about platelets? They are responsible for clotting of blood. Okay. Platelets are responsible for clotting of blood. Remember that, it's very important, and I'll write the name of all of them over here so that you do not have any confusion later on. So erythrocyte, they are RBC, that is red blood cell. It can be C also. Leukocyte is WBC, and the most important, thrombocyte is your platelets. So yes, thrombocytes are platelets, so you have to remember the names of them as well. Why? You'll get to know right now. So these are the blood cells, children. I'm not going in detail because we do not have much amount of time. So this is the reason why I told you to remember the names of the blood cells. So there is some fluid which is known as lymph fluid. What is lymph fluid and how it is useful? I'll tell you. So basically, when blood comes from the, okay, I can make it here itself. Okay, so when blood is coming like this from the arteries, it going through the arterioles and then to the capillaries, what happens is that if I zoom this particular structure in, there will be blood, as I said, in the capillaries, there will be just one cell thickness, right? Please remember, it's just one cell thick, okay? And over here, you will be having pores. Over here, you will be having pores. So basically, with the pressure that the blood is coming, lot of things, for example, the plasma in which the blood is floating, I didn't tell about that. So basically, there is plasma, the yellowish thing which is there, okay, in which the plasma is the thing in which the hormones, the proteins, the gases, a lot of things are floating in there, and in that only the blood cells are also floating. So 55% of your blood is plasma and 45 basically calculate is the blood cells, okay. So too much amount of plasma is there which helps in the easy movement of the blood cells. Now the plasma, as I said, will be having dissolved proteins, hormones, lot of things. So this plasma is there which is coming along with the blood, obviously. So the plasma basically oozes outside. Can the RBC go outside, children? Can the RBC go outside? RBC does not go outside, okay, because it's kind of big in size. Your platelets, thrombocytes, do they go outside? No. Platelets also do not move outside. What is that something that moves outside? Your leukocytes. What are those? Leukocytes or WBCs, they can change their shape, which is why they can easily move out. So plasma has moved out, leukocyte has moved out, and also certain proteins and things like that has moved out. So over here, when you talk about lymph, I can draw it here and show you what. So this is the blood vessel from which what all has moved out? Plasma has moved out, WBCs have moved out, that is lymphocytes, okay, and proteins have moved out, certain proteins, smaller ones, okay, amino acids, some things like that, okay, and then pathogens. Pathogens also move outside. But where is it moving outside? All these capillaries are going to different regions, right, between the cells. So basically, these things are getting moved out into the intercellular spaces. What do you see? This space is known as interstitial space or intercellular space. So I am writing interstitial space. It is moving around over here. Now, if the fluid accumulates there, then that will be a problem, and also it will cause inflammation, right? It will cause inflammation. That is one big problem. Apart from that, there is one more big problem. They are making the blood pressure less. You have to take all these things back. You need them. Pathogen is not needed, rest everything is needed. So basically, there are another set of blood vessels, not exactly blood vessels, I cannot call them. They are known as lymphatic capillaries. They are known as lymphatic capillaries, which takes these fluid inside them and then opens into lymphatic vessels. The lymphatic vessels have regions which are known as lymph nodes, which are rich in more amount of WBC, which could fight against the disease, and then the lymph directly opens into a vein, which will now open into heart. So whatever things got loosed out, it is drained back and taken back to the vein. Okay, it is going up. I didn't have area to make it, that's why I am making down. Okay, so basically, this fluid forms the lymph fluid. It's getting inside the lymphatic capillaries first and then the lymphatic vessel. Now over here lymph nodes are there which have high amount of WBC. This WBC captures the pathogens which also comes in. And then everything clean, everything goes to the vein, then to the heart, and the whole process goes on. Okay, so lymph, also known as lymphatic tissue or lymphatic fluid, is a collection of extra fluid that drains from the cells and the tissue. The other substances include proteins, minerals, fats, nutrients, damaged cells, cancer cells and foreign invaders as well. Now children, listen to me. As I said, blood vessels, that is our arteries and veins, cannot take in fats because fat is very big. But lymphatic vessel, it definitely takes in fats as well. So for absorption, remember in small intestine I was saying there was a lymphatic vessel, it is taking in fats. Lymph is colorless fluid containing of plasma and leukocytes and not erythrocytes and thrombocytes. Please remember, it is very important. Okay, what is the function of the lymph? It maintains the fluid levels in the body. Okay, it absorbs the fat, it protects the body and transports and removes waste products as well. Then difference between blood and lymph. If its plasma has more proteins, calcium and phosphorus, obviously not everything can move out. Bigger proteins will still remain in the blood itself, but lymph will be having lesser protein, lesser calcium and phosphorus. It consists of plasma, erythrocytes, thrombocytes and leukocytes. It does just have plasma and leukocytes. It's red in color due to the presence of hemoglobin in RBCs, whereas it is colorless because hemoglobin is not there, RBC is not there, right? So yes, it is colorless, please remember it. Go ahead, over here we have transportation in plants and we are going to end the chapter really soon. So yes, transportation in plant, it involves xylem and phloem, which are the vascular tissues that is present in the plants. Now there are two theories which is responsible for the transport of water through the xylem. So xylem is responsible for xylem is responsible for transportation of what, children? It is responsible for transportation of water, and phloem is responsible for transportation of food. So for phloem, for food. Okay, now going ahead over here, we have two theories as I said over there, root pressure theory and transpirational pull theory. What happens in the roots? I'm just telling it very fast, okay? In the roots, you need obviously solutes, that is need of solute. So ions basically from the soil get inside the roots with the help of active transport, as in you are giving off energy to take in things. You are giving off money to take things in. So it is very much important. Nutrients are needed, which is why ions are taken in. Now when ions are taken in, obviously the amount of solute in the roots will be very much. To combat this, water molecules will be now moving from outside to inside with the help of osmosis. Okay, it will be moving down the concentration gradient, right? Will be moving to into the roots. Now what will happen when water molecules basically gets accumulated in the roots? They are going to push each other up. Okay, this increases the pressure. This pressure that is there, which is known as root pressure theory, it is a pushing force, which is why water is getting moved up, and we want that. We want the plant parts to receive water. But again, this particular process would not be very much efficient, because it can take things, but it's going against gravity, so it is a big trouble. Now second thing is transpirational pull theory. So there is a pulling force over here. Let's see how. So when evaporation happens, water is lost in the form of vapor, and this process is known as transpiration. Okay, so more water molecules are pulled up. What happens when water molecules goes out, and these water molecules, no children, they are like this. They hold each other. Okay, there is another water molecule, it is holding each other. So when one is going up, the other one is being pulled up. They are not leaving their hand. So this force which is there between H2O and H2O, that is similar things, it is known as cohesive force, which is why when water molecules are lost in the form of water vapor, the other water molecules is pushed up. So this is a pulling force. Okay, pulled up, not pushed up. It's a pulling force. And also one more thing, what happens is that this water molecules is having a bond with over here also, a wall also. This force is known as adhesive force. So basically it is moving like this. Okay. In the form of different, like not Hrithik Roshan, different. So I hope you got this. This is transpirational pull theory, and this is root pressure theory, with the help of which water is getting transported. You have the PDF, you can have the PDF, you can write your notes properly. Going ahead, translocation in plants, that is when food is needed to be transported in plants. This is a bit more complicated but very easy. Translocation is the process by which assimilates from photosynthesis. What do you mean by assimilates? Basically, whatever important nutrients are being made with the help of photosynthesis are moved around the plant from where they are made. So the place from they are made, it is known as source, and from there where they have to be moved or where they are needed, it is known as sink. So movement is from source to sink. Where is it from? Source, from where they are made, to sink, where they are needed. Now this movement, children, it is bidirectional. can move up as well as down. It's not like the upper part of the body will be only needing food and the lower part won't be needing food. It's not like that. So it is bidirectional, whereas in the case of this thing, in the case of transportation of water, it was unidirectional. The water was going from the soil to the up. That is why we say soil, you have to put water in the soil, not over the plants. Okay. So water will be going always in one direction, that is up. Yeah. Now next thing, plants convert the sugar made during photosynthesis to sucrose. So usually we know that the plants produce something that is known as glucose, right? But this glucose can't be transported, so which is why this glucose is converted to sucrose. That means for glucose to getting converted into sucrose, you just add one more sugar, that is fructose. If you are not able to remember fructose and all, just remember glucose is getting converted to sucrose. Why? Because if glucose is the guy that goes around, it could go react with lot of things and cause lot of trouble. We want glucose to be in glucose form, which is why it is transformed into some other form, which is a non-reducing sugar. It won't be undergoing any heavy chemical reactions, which is why this conversion is very much needed. Please remember it, sucrose is the one that is getting transported, not glucose. It has come so many times. Now if you talk about phloem tissues which are responsible, they are made up of two main type of living cells. One is seed tubes and the other one is this thing that you see over here. It is the companion cell. So this particular thing that you see over here, it is the companion cell. Companion cell, no, it has the nucleus and everything. Sieve tube element just has the cytoplasm, little bit amount of cytoplasm, that's all. They do not have nucleus. So companion cell is the one that provides everything to the sieve tube. Now, companion cells are linked to the sieve tube members by processes which are known as plasmodesmata. As you can see over here, if I try to zoom this in a bit, you will be able to see this. They connected through pores which are known as what, children? Plasmodesmata. Okay, I'll tell you why it is needed. And companion cell also has many mitochondria which could later help in active transport by contributing ATP. So some active transport is going to come. Active transport as in you are going to give energy for transporting things. So let's see, these are the steps involved. So over here, this is the place. Suppose this is palisade mesophyll. Okay, so I'll just explain this. Over here the food is made. The food is converted to sucrose. From here it is transported to companion cell. Okay. From the companion cell, things are being transported to this particular thing, that is what? Your sieve tube cell. Okay, sieve tube cell. So just imagine that already sucrose is there. Still sucrose will be transported from here to here because food is needed anyways. Okay, and that pressure has to be needed there. I'll tell you why. So hence, this transport that you see from here to here, it is active transport. That means ATP is being required. You are going against concentration gradient. Already there is something. You are making things more populated there. So this is active transport where you are utilizing ATP for transportation of the things that is present over here, that is sucrose, into the sieve tube element which already has sucrose. Okay, so this happens. Now the next thing that happens is basically when solute is too much over here, parallelly there will be xylem over here. Xylem sees that there is too much amount of solute, I should probably give my solvent, that is water. So now the xylem gives water to this particular region. Now you only think water is there, this sucrose is there, everything is there. Won't it create a pressure difference between two regions? So over here everything is there, over here nothing is there. So wherever the things are being low, slowly it will be moving to that particular region. This is known as mass flow. So this movement of water would lead to increase in pressure in the sieve tube cell and further would lead to mass movement of the phloem sap. This whole thing is known as phloem sap because it's there in the phloem. Okay, to the area where the pressure is low, and this flow is known as mass flow. Okay, now this is happening in the source. This was the source region. I am telling you again, things are made, put it in the companion cell, again active transport is having, already things are there, but still you are putting because you want things to get transported. If solute concentration is not high here, water won't be coming here. If water is not coming here, mass flow won't be happening, which is why everything is happening. Okay, now in the sink what is happening, children? So suppose this is sink region, the sucrose then moves. So you know the concentration of sucrose is less here because it is needed over here. Sucrose moves outside and the water molecule is like, hey, I came for you, you're going out. So like, yeah, I'm going out. So water molecules be like, I'll be going back to xylem as well. So basically it's not like that. It's not like that, but the xylem will be having water along with certain nutrients, and here the water will be there. So basically, if you see again, the concentration gradient will come back over here. There will be too much amount of water over here. There are nutrients over here along with water, which is why water comes back. You can remember like that as well. Okay, so it has given everything. Okay, it has given everything to the sink region. Now the water moves out to the xylem, and the same thing repeats. It is bidirectional, can go up or down. Done. Please go give a read once. Okay, what are the following transport? Xylem, it is transporting water. Phloem, it is transporting food. Pulmonary vein, tell me pulmonary vein in the comment section. Pulmonary vein, what is it transporting? Oxygen, oxygenated blood or deoxygenated blood? Only exception is there. So it has to transport what? It has to transport oxygenated blood. It is the only exception of a vein. Vena cava, deoxygenated blood. Pulmonary artery, again an exception, deoxygenated blood. Aorta, oxygenated blood. It's the largest artery. So the given figure represents the movement of water and minerals in xylem and movement of food in phloem. So this is one thing and this is another thing. We have mentioned X, Y and Z. Let's see. Choose the correct combination. X is this X, okay. Major conducting cells in xylem. It's not xylem. So wherever you see xylem, you cut it out. So this is xylem, this is xylem. This is also xylem, so you also cut it out. So everything is cut out. Let's see if this is correct. So major conducting in phloem, okay. Now Y is nucleated. Yes, companion cell has nucleus. Done. And Z is unidirectional. So it is B option. See, that's how you need to find the answers. Okay. Now going ahead, explain the process of transport of oxygenated and deoxygenated blood in human beings. You have to make the diagram, make a flow chart and show it to them that this is how, start from lungs and say that this is how it happens. It's a 5-mark question, so you have to make the flow chart, explain in detail that this is one tricuspid valve, everything is involved, pulmonary circulation and systemic circulation is involved. If you are not mentioning about the valves, then please mention about this as well. Okay, anyways, you have to mention about double and the single circulation and this, why it is important and everything, whatever is possible, but you have to explain to them. Okay, so what are the things that we need to tell? First thing, flow chart. Give the flow chart that we have. Okay, and then explain in detail how things work. Talk about systemic and pulmonary circulation as well. Okay, will you be able to do that? And explain the importance, why oxygenated blood is needed. Okay, since it is asking about human body. Fine. Going ahead, give reasons. Ventricles have thicker muscular walls than atria. Tell me why ventricles? Because ventricles are the one that is putting pressure to get things out. So ventricles will obviously be having thicker walls. Transport system in plants is slow. Why is transport system in plants slow? Obviously they do not have organ system and anything for that. Okay, it's just phloem and everything. It's all based on the sap and the movements of the active transport and the passive transport. Whatever is happening, it's diffusion that is happening. So which is why the transport system in the plants will be definitely slow. Circulation of blood in aquatic vertebrates differs from that in terrestrial. You know that in aquatic there is less amount of dissolved oxygen. During the daytime, water and minerals travel faster through xylem as compared to the night. Okay, yes. So at night, okay, so during daytime transpiration is happening. This is a very important question. Okay, during daytime transpiration is happening, but at night time, so during daytime transpiration and root pressure pull is also happening. Root pressure push is also happening, but at night is just root pressure theory that works, which is why at night transpiration cannot happen, right? Which is why that pull is not being created. Veins have valves, whereas arteries do not. You know the answer, because veins can show backflow of blood. There can be backflow of blood. Going ahead, last topic, excretion, and then everything is done. So this is a structure of a nephron, children. I will label one by one. Okay, so there will be a renal artery that comes in, that branches off for different, so this is a renal artery. Okay, and wait, Yeah, it branches off. This thing that you see is afferent arteriole. Please remember it, it's important. Afferent arteriole, A for coming, RI. Okay? Now going ahead, this is efferent, it is exiting. Efferent arteriole as in exiting. Okay? But what is it exiting? Basically, this afferent arteriole comes and forms a group of capillaries there. Okay, it forms a group of capillaries there, and this bunch of capillaries that you see over here, it has a specific name. It is known as glomerulus. Glomerulus. Okay, and this cup-shaped thing that you see over here, it is known as Bowman's capsule. Okay, now going ahead, I'll just talk about the structure first and then we'll talk about the function. This thing, that is coiled structure that you see over here, okay, and it is very proximal to the whole structure, that is Bowman's capsule. You call it proximal convoluted as in coiled, tubule as in tube. It is proximal, it is near, that is why it is proximal convoluted tubule. Now this is this structure that you see over here. This is Henle's loop or loop of Henle. Please practice the diagram. Okay. Now this structure that you see, it is distant but still coiled, so you call it distal convoluted tubule. Okay. Going ahead, what do we have? This thing is the collecting duct. Now children, please think, this is a nephron, this is one single unit. Okay, this is one single unit for excretion. So basically there will be lots of collecting duct. They will open up into something like this. So I have to go like this. So this is your kidney, which will be having so many nephrons. So many nephrons. This is the kidney, right and left one. Yeah. And now there will be something like this that comes from each side of the kidney. This something is known as ureter. Okay. Over here, they will open up in the urinary bladder. Okay. And this is urethra, that is the opening of the urinary bladder. Now, there will be cortex region. This region is known as cortex region, and this middle region is known as medulla of the kidney. Why it is important? Because half part of the nephron, so there are so many nephrons around here. Half part of the nephron is in the medulla and half is in the cortex. You will get to know that. What is kidney responsible for? Excretion for sure. You can take a screenshot of this particular slide, okay, because it's not there. I have added this slide now. It might be not there in the PDF, okay, so please do that. Okay, what is kidney responsible for? Primary function is what? Excretion. Okay, maintaining the level of fluids, maintaining the level of water and ions, everything. It is also responsible for formation of RBC. Why? Because the kidney is going to release, helps in RBC formation as kidney erythropoietin. So there is a hormone known as erythropoietin which is being associated with kidney, and if it is not there, RBC won't be forming. Next thing, vitamin D formation. It's not like vitamin D forms here. There is a conversion of vitamin D that happens, a stage of that happens here. If vitamin D is not there, calcium absorption won't be taking place. Calcium absorption. So basically it is linked with that. So all these features are extra, but you need to know. So a person who do not have a kidney is surviving on dialyzing machine, so these things would be definitely hampered. Even though waste and everything will be removed, these things will be definitely hampered. You should be knowing that, which is why I told about this. Okay, so kidney is not the only excretory organ. There are secondary excretory organs as well. There are secondary excretory organs as well. What are these secondary excretory organs? Liver, lungs give out this thing, no, and skin. Lungs give out carbon dioxide, skins give out excess water and salt, and liver also converts toxic things into less toxic forms. Okay, and I'll tell you one more thing. There are different organisms. A monotelic. So ammonia is the most toxic form of nitrogenous waste. So our body is going to remove nitrogenous waste because nitrogenous waste is very common and it has to be released. So ammonia is the most toxic form. In our body also ammonia is being released, but in our body liver is going to convert this ammonia into less toxic form, that is urea. Remember, we are having urea, not uric acid. That is different. Okay, so there is immunotelic organism. So for making something less toxic, what do? You put water so that it becomes dilute, right? So these organisms, organisms who release ammonia as a waste, they will definitely have lots and lots of water around them. So basically aquatic animals will be having a monotelic mode of excretion. Ureotelic is showed by us, humans, and adult amphibians. I'm not writing it over here. Now uricotelic, as in uric acid is being produced. Okay, the least toxic form, if you see the least toxic form is uric acid, and it is released. It does not need too much amount of water to be lost. So uric acid is being released by, you have seen this guys, birds, okay, reptiles, they all show uricotelism. So remember, in our body, in the liver, ammonia is converted to urea, and this urea is something that has to be excreted out. This ammonia, if it sustains, it is very toxic for us, which is why it is getting converted to urea. I hope you got this. So these are also secondary excretory organs. These are organs, secondary excretory organs. Please remember that. Now going ahead with this. So we have all this thing that is present over here. Now let's read it one by one. Blood comes from the renal artery. There is renal artery, there is renal vein, there is renal artery. Renal artery brings the blood and veins takes the blood away. Okay, remember, our right side of the kidney is a little bit in the lower position because of the liver being there at the back. Okay, not at the back. Basically the kidney is towards the posterior side, that is towards the back side, but because of the liver the position is different. So your right kidney would be little lower than the left kidney. Okay, now so blood comes from the renal artery and moves through the efferent arteriole. So this is the efferent arteriole, it moves like that. Because of the pressure, certain components present in the blood moves from glomerulus to Bowman's capsule. So basically just imagine too much amount of blood is coming in too much pressure. So a lot of things could move outside into the Bowman's capsule because they are forming a bunch of capillaries over here, right? Remember the name of the capillaries? You call them glomerulus. So a lot of things are coming inside. What are those things? You have to remember: ions, water, urea, that is our waste, glucose and amino acid. Proteins cannot come because proteins are big, so amino acid comes in. Ions can come, okay, water will definitely come. Urea, which is our waste, it has to go, it also comes. Glucose also comes, okay. All this together you call them as filtrate. Okay, you call them as filtrate, and this process is happening in the glomerular region, which is why it is known as glomerular filtration, or you also call it ultra filtration. You call it glomerular filtration or you call it ultrafiltration. Note, only small components could pass through, not bigger. That is why blood cells, none of the blood cells pass through. Okay, please remember, none of the blood cells pass through. Over here you have iron, water, urea, glucose and amino acid. If blood cell is passing through, then that's a problem. This is the first step of urine formation, and the urine formation has come so many times. So steps of urine formation: first one, glomerular filtration or ultrafiltration, where things moves from the glomerulus to the Bowman's capsule. What are the things? Ions, water, urea, glucose and amino acids. These are known as the filtrate. Going ahead, now things got filtered, but our blood is obviously going back Okay, you have the efferent arteriole which takes the blood. So blood continues to flow from efferent arteriole to glomerulus to efferent arterioles. Okay, and it goes on and it forms, these efferent arterioles are now going to form capillaries around all the nephron body. Okay, this efferent arteriole is now going to form the capillaries around it. Let's see. Next step is selective reabsorption, for which we needed the capillaries, which is why I mentioned that now. So most of the component in the filtrate are selectively reabsorbed. Selectively reabsorbed as in meaning selecting things and reabsorbing things. Okay, not everything is being selected. What is being selected? Ions, water, glucose, amino acids. So lot of things, as in half of it is gone. Just urea is left because urea is the waste. Okay, but to our knowledge, let me tell you, little amount of urea is also taken back. So this process is known as selective reabsorption. But ma'am, why is it happening? So the thing is, it was because of the pressure that things moved down. It's not like the body didn't want it. So body still wants it, which is why things are being taken back. But where does the most amount of selective reabsorption take place? In the proximal convoluted tubule. This thing that you see over here, this is the place where most amount of selective reabsorption takes place with the help of the capillaries that surrounds them. Okay, going ahead, what do we have? Blood flows through the capillaries that wraps around the nephron, and while all this happens, there are so many other things happening in the background. So blood is flowing, capillaries taking in, things like that is happening, but there's other things also happening in the background. Let's see. First thing, tubular, did I miss something? Okay, now the tubular secretion. One more thing is there. First one was glomerular filtration. Second is selectively you're taking back things. Now there is one more chance that the nephron is giving to the blood. If you want to give me anything as of now which has to be excreted, give it to me now. This is what you mean by tubular secretion. So from the blood, certain components which are high in amount in the body, or if it is of no use, then moves back to the nephron, and this is known as tubular secretion. Even though tubules are not secreting, but still the name has come like that. Please understand, this is tubular secretion, where initially what happened, things came into the nephron, then things went out to the nephron, now things are coming back to the nephron. You can think like that. Okay, what are the things? Something that is in high amount in our body, or probably is of waste. So what all moves back from the blood to the nephron? Certain ions which could be increasing the pH of the blood. If pH of the blood is increasing, then that's a problem. We have a normal neutral pH, so it has to be maintained. Or certain drugs. Okay, certain drugs, medicinal drugs, you know, they also might be there, which has to be excreted out. So all the things are being taken back. So these are the three steps of what? Urine formation. And at last, what do you get, children? Whatever thing is there, everything moves to the collecting duct, and then to the ureters, and then to the urinary bladder, and then goes out. Okay, so these are the three steps: glomerular filtration, selective reabsorption, and tubular secretion. Okay, you have the notes, please read it, but I have to tell you one more thing. That is, now this particular region that you see, children, it comes under cortex. You remember, in the kidney there was cortex region, and then there's the inner region, that is medulla, right? So basically over here, this comes under cortex and this comes under medulla, and here they're saying, hello, I am this is descending loop of Henle. I am permeable to water but not permeable to solutes. So basically water can move out, but whatever solutes are there, they cannot move outside. That is about descending loop of Henle. Now that is something extra. If you want to know, you can know. Next thing over here, I am ascending loop of Henle. I am not permeable to water, but I am permeable to solutes. That means solutes can go out, but water cannot. So that is two separate scenarios. Why is it important? Let's see that. So ions move out by active transport. Hence, in the medulla, the solute concentration is more now. So basically, ions which was there, it can obviously move out. It is a solute, right? It can move out through ascending loop. So ions, they move out. Now, when the ions, they move out with the help of active transport. They are expending the energy to move out. Now, you might be thinking why they are using their energy to move out. The thing is, as solute concentration is more, hence the water from the loop of Henle will also come out from the descending loop. Now, in this way, you are not losing the amount of water. Your urine will get concentrated and it will go out. That is the whole purpose of doing this. Again, I will repeat. So this side is okay for a solute to move out. This side is okay for the water to move out. So solute move out with the help of what? Active transport. Okay. But what about here? This side, basically when solute is too much here, water moves out. Hence, it concentrates the urine. So you might have seen that the initial filtrate, okay, initial filtrate was 180 liters. The initial filtrate which is being taken up in the nephron, it's 180 liters, but then out of this we urinate only 1 to 2 liters daily. That means everything else is taken back. Everything else is absorbed back. This is very much important for a healthy kidney function, and this is known as glomerular filtration rate. Okay, will you be able to remember this? So I put so much effort into making you understand this. I hope it is really helpful for you. And as I said, please remember the names of all these things. Okay, do not forget, no big things can come in, so no blood cells can come in. If blood cell is coming in, that's a problem. Now there is a situation given in your NCERT where a person, where people who do not have proper functioning kidneys, they opt for hemodialysis. Okay, they opt for something that is known as hemodialysis. What exactly happens? It is also known as artificial kidney. It is known as artificial kidney. Okay, so what exactly happens in there? So blood, will be a connection with your arteries so that the blood could be taken in. The blood moves in. There will be heparin injection. Heparin does not allow the blood to clot, so it is an anti-clotting agent. Okay, and now it moves to the dialyzing machine. So over here in the dialyzing machine, no children, it looks like this. There will be certain tubes, and these tubes, no, they are semi-permeable. These tubes that you see, it's what? Semi-permeable. It is made up of nitrocellulose. It is made up of nitrocellulose. So basically what happens, okay, and there will be fluid over here, which is the dialyzing fluid. So doctor checks all of your ions and whatever is there and see what is the condition of your body. According to it, this dialyzing fluid is made for different people. Okay, so over here there is one thing that will be in less amount, or something else also if it is needed to be removed from our body, and that's something that will be obviously in less amount will be urea. Will be what? Urea. If the concentration of urea is less over here, when your blood passes through and blood has urea, obviously it will move out. Agree? If the concentration of urea is less in the dialyzing fluid and it is more in the blood, the urea will be moving out, and also certain ions will be moving out, which is not important. Okay, sometimes even if it's not of our will, things could move out, and that is a problem, which is why it's a problem. Obviously you need a proper working kidney as well, right? So this is dialyzing fluid, and this dialyzing fluid has to be changed, or else what will happen, the concentration of urea will be more here, so it could go back, right? So later on, this particular thing, okay, the fluid is being taken back, new fluid is come, and now the filtered blood is taken back again, and this is connected to the vein of the human being. This vein obviously will be tracking things to taking back to heart, and the same thing repeats. This is done in a week almost thrice. Okay, and as I said, so people who have doing hemodialysis, or who are doing hemodialysis, they will be obviously getting their blood filtered. Thus, what is that one thing that does not happen, or two things? Erythropoietin is less. So in them, the blood cell formation, red blood cell formation would be obviously bit altered, and also vitamin D formation would be altered. Okay, so that is about hemodialysis, and please remember 180 liters of blood, and this much is being removed, only 1 to 2 liters. Okay, and all the names of everything that I have mentioned over here, glomerular filtration, tubular secretion, and everything. The structure of nephron is very much important. In the case of plants, how does excretion happen? I didn't put it over here, I'll just tell you. So basically they have gums, they have resins being released, okay, or else they shed off their old leaves, which will be having all the toxic chemicals. So that is how they give out their not so important things. So I think that is done. Yes, Let's do. In our body, which organ is responsible for conversion of ammonia to urea? I explained to you, right? It was what? Liver. It is what? Liver, which is responsible for conversion of ammonia into urea. Name 4 types of metabolic waste products produced in humans. So, urea. Then what do we have? Carbon dioxide. We have sweat. And then anything that you wish to put. Some toxic salts. Some salts that you put. And always remember, in our body, the most common waste is nitrogenous waste, and we are ureotelic. Name any 2 excretory organs other than kidney. You know that. The board mein it came. What is the excretory organ? You have skin and lungs and liver. Now children, if you want the PDF for this, again I am telling you, for those who didn't attend it, listen to me carefully before, you can find it in the description box. You can click on the link. When you click on the link, it will lead you to some page like this, where it is date wise given that what will be taught. PDF for the link, a link for the PDF will also be given. You can click on that and get the detailed PDF. Will it work for you? Yes. And this is the homework that I want to give it for you. You can take a screenshot if you want to try it right now, or obviously you can go, if you had the PDF, then what to worry. If you know the answer for this right now, you can obviously put down in the comment section. So this is one thing that I have. This is the second thing that I have. Third question, and then there is a bit long questions as well. So you can 2 marks, 3 marks and 5 marks. Accordingly, you can try to frame your answers. In this way, you will be getting a good practice. And if you're just learning without practicing, then that is a huge trouble, which obviously you should be avoiding. Okay? So please do these questions. And yes. Now, if you're confused, children, regarding what stream you should be choosing next, whether it should be medical or engineering or some other field, whatever is it. So we at Diksha are providing DSAT, so Scholastic Aptitude Test, okay, where there will be 60-minute concept based lecture, after which there will be an aptitude test, there will be one-on-one counseling, and then they will be telling you what could suit you better. Okay, so for that, again, if you want to have some information regarding this, and there is 90% scholarship as well, so you can again find the same link in the description. Okay, click on that to get to know more details. So yes, champions, we completed life processes, and if you were with me throughout the whole process, you completed it just like how I taught, then give yourself a big tap on your back. You did a really great job, and you are going to score really well for your midterm. All the best for your boards, and have a really good day. Do not forget to smile. Take care. Bye-bye. Also, do not forget to subscribe to our channel, that is Diksha Vedantu Foundation. So stay updated. Bye-bye.